This is a production of Cornell University.
So, good morning, everyone. Welcome to Plant Science 4950 Senior Capstone Seminar Symposium. Mike Scanlon and I are thrilled to host everyone today. I am Chris Smart, one of the co-instructors for the course, and I will be the official timekeeper, keeping us on time because we know people are going to hop in and off the Zoom today. Mike, you want to introduce yourself and get us started?
Hi, I'm Mike Scanlon, plant biology. I co-teach this course with Chris, as you just said. Yeah, we have a great lineup today, so I think we'll just get started. Our first speaker is Marlie Lukach. She's an Ag Sci and International Agriculture and Rural Development major who's concentrating in sustainable cropping systems. After graduating, she's planning on staying for a PhD right here at Cornell in plant breeding with Mike Mazurek. And the title of her talk today is The Future of Carbs. If you can share your screen, Marlie, we can launch this.
All right. Good morning, everybody. So I want to start you guys off with a question. How many people listening out there like bread or other carbs? You can raise your hand. All right, I see quite a few hands out there. So statistically, 5% of you carb eaters have some sort of wheat sensitivity. And the biggest culprit for these wheat sensitivities are ATIs. And ATI stands for alpha amylase trypsin inhibitor. And these wheat sensitivities range on a pretty broad spectrum, which can go on the low side of issues, which you might just have some stomach discomfort when you eat carbohydrates, all the way to having pretty severe allergic reactions. And through evolution, ATIs have been an insect defense mechanism. So basically, when the insect nibbles on the kernels of wheat and ingests the ATIs, it inhibits digestive enzymes from breaking down starches into maltose or other simple sugars. The insects can't obtain nutrients from the food they're eating and they die. So it's a pretty great mechanism for wheat and other small grains. Problem is, you still have these issues when people eat small grain products. But there's hope for people who have these wheat sensitivities because ATIs are readily used in the beginning of germination. So as part of my honors thesis, I looked at how we could use germination as a pre-processing technique to reduce levels of ATIs. So what we did was we took 2 different wheat cultivars and we did Petri dish germinations at several times temperature and pH values. And we used this DNS technique, which uses 3,5-dinonitrosalicylic acid. And basically, this acid creates a color indicator. So when you add your enzymes to the wheat sample, the more maltose starch is broken down into maltose, the darker your sample is. So over here in this picture, we can see this darker sample has more starch broken down to maltose than this lighter color sample over here. And we can take these colored samples and indirectly measure maltose using light spectrometry. And we can use this equation here, putting in our absorbance reading, and we get the maltose extracted, and we can indirectly know how much ATIs are left in the sample depending how much maltose was released. So more maltose means less ATIs in the sample, better for us. So when we first looked at time, we looked at a range of periods from 12 to 48 hours. And what we found was there's this kind of sweet spot where you get a high level maltose release right here at the 36 and 42 hour time point before you start getting the presence of shoots and roots, which decreases the quality of the grain to be used in other products. So we found that overall, we like this 36-hour time point for having low amount of roots, but having a high enough release of maltose that we know we're getting our ATI levels low enough that it's not going to cause problems to people with wheat sensitivities. And then we looked at the effect of temperature. So we looked at 20, 30, and 40 degrees Celsius. And what we found was at 20 degrees Celsius, which was about room temperature for us, in the laboratory, even though there was quite a bit of variability here in the maltose release, overall maltose was significantly higher in all the 20-degree samples. We also found that at these 30 and 40 degrees temperature samples that we had a lot of mold and issues with germination, which obviously when you start having low germination and you start having mold and things, we can't use these products, these wheat for other products. So overall, we decided that this 20-degree temperature point, was the best for germination. And lastly, we looked at the effect of pH on maltose release. And what we did here was a Tukey test. And basically, what a Tukey test does is it takes your measurement for each variable you're looking at, and it tells you if they're significantly different from each other. And it gives you a letter that corresponds to a significant difference group. And what we found here is that these acidic pHs of 3, 4, and 5, we did not see any significant difference between the maltose release, as we see all 3 columns have an A. So at least at these pH ranges, we didn't really see an effect from pH. So our conclusions from this part of my study was that the optimal germination conditions seem to be about 36 degrees, or 36 hours at 20 degrees Celsius. pH does not seem to have much impact on germination and maltose release. And there's possibility for future food science and human nutrition research to use this germination as a pre-processing step to make wheat products for people who have wheat sensitivities. And lastly, I would like to thank Cornell University's Small Grains Breeding Program for helping me with this project, and a special thanks to Dr. Mark Sarles, James Tanaka, and Daniel Sweeney for helping. Thank you. Are there any questions?
That was a fantastic job, Marlie. Really well done.
Well done, yes. Great stuff.
And thank you so much for leading us off.
Okay, our next talk is from Rosemary Glos, a plant science major with a minor in botanical illustration, and her concentration is evolution and systematics. After graduation, Rose will be taking a gap year before entering a graduate school of her choice, probably. And then today her title is Investigating Leaflet Anatomical Variation in the Leaf Cycad genus Zamia, I should say.
Great, thank you very much, Mike, and thank you everyone for tuning into the symposium today. Today I'm going to talk about my senior thesis project in the Spec Lab, where I looked at leaflet anatomical variation in a rather charismatic group of cycads. To start us off, I'd like to just remind everyone of what exactly a cycad is. I would hate to think anyone had forgotten, but just in case, cycads are gymnosperms, so they fall in the tree with other plants that produce seeds, but not flowers, such as conifers and ginkgo, etc. And cycads have the unfortunate distinction of being the most threatened order of plants on Earth. And because of this, or maybe despite that, we have some surprising gaps in our knowledge about cycads. And whenever we don't know enough about something basic as anatomy or morphology for a group of plants, we have a hard time defining species and therefore understanding how many species we have to conserve. So for my own project, I actually had 2 goals, a scientific one and a personal one. The scientific one is the one I'll focus on today, and that's just filling in a gap in our knowledge of cycad anatomy. But I don't want to neglect the personal one. The personal one is for me as a senior, hoping to go to graduate school soon, to design, implement, and communicate some original research. And that second goal is just as important and perhaps even more important than the first one. So what did I do? Well, I'd like to frame my research question by speaking specifically to the resources that have been available to me, because this project would not have been possible without them. So in the center here is my research question. It asks, are anatomical characters phylogenetically informative in the genus Zamia? I'm focusing on the genus Zamia because it's the largest cycad genus in the New World, and also it has a surprising paucity of anatomical information in the literature. And so by asking whether we can use anatomical characters to define clades within the phylogeny, we have a better idea of how many species there are, how they're related, and therefore how we can conserve them. In order to ask this question, I need a phylogenetic framework. And fortunately, I have a newly published phylogeny found out by Michael Colonje and his collaborators. So I have the framework I need. And then I also need the actual plant material to compare it to that framework. So in January, I went down to Coral Gables, Florida, and I collected material at the Montgomery Botanical Center and Fairchild Tropical Botanic Garden. And then, now that I had the material and the framework, I needed the means to actually section, image, and prepare all of those slides. And for that, I had an in-house anatomy collection and lab, and that's Alejandro Gandolfo's lab and the Cornell University Plant Anatomy Collection. Lastly, I needed expertise, because when I started this project, I didn't actually know how to do any of these things. I didn't know how to properly prepare slides or really look at an anatomical section. So I'm very thankful to all the members of the Spec Lab, in particular Chelsea Speck and Shayla Salzman, and also Barry Tomlinson, an anatomist at Harvard, who was absolutely instrumental in helping me get started. So I would love to have sampled and examined every single species in this genus, but it's just not tenable. Many of them are not in cultivation, and even if they were, I just didn't have the time, I'm afraid, to look at every single one. So instead, I chose 20 out of the 81 Zamia species, plus an outgroup, that have fairly good, broad phylogenetic and geographic distribution across the genus. The circular phylogeny on the bottom here shows the samples that I chose in green, and as a whole, it's the whole phylogeny of the species. So this just shows, of the genus. So this just shows that the ones I sampled have pretty good representation across the major clades. On the right here is a pared down phylogeny showing just the species that I chose, along with their geographic distribution. And this distribution closely matches that of the genus as a whole. I don't have the time, unfortunately, to go into all the details of what I did to prepare my samples, but it fell into roughly 4 categories. I started by collecting all of the samples down in Florida. I collected individual leaflets off of larger compound leaves, and I fixed them in FAA and stored them in ethanol. And then back at Alejandro's lab, I sectioned them on a rotary microtome. I stained them with a wide variety of stains that specifically elucidate certain characters of the anatomy, specifically safranin, toluidine blue, Sudan 4, and others. And then I examined them under a compound microscope and scored them for a wide range of anatomical characters that varied across the genus. And so today, I'd like to just show you some of my initial results, starting with what the leaflets look like as a whole. So this circular phylogeny here shows nearly all of the species in the genus. These were all of the ones I was able to find down in Florida. The main gaps are predominantly Amazonian species that have only been recently described. And all I want you to take away from this giant graph is that there's a whole lot of variation in this genus. The leaflets are thick or thin or long or short, some are variegated. But the thing is, if you look at it, they don't cluster in with similar characters. So it's not as if all of the long, thin ones are in one clade, or a really good one to look at is the plicate leaves, the ones that are folded, and they don't all fall together either. However, there has been some other work showing that anatomy could potentially be more informative in the genus. And so there may be anatomical characters that correspond to certain clades. So that's what I looked at. And in order to show you this, I'm going to walk you through what a general cross-section of a Zamia leaflet looks like, just in case it's been a while since you've looked at an anatomy slide. So this is a cross-section of Zamia boliviana, stained with safranin. And I'm just going to walk you through what we're seeing here. So below the cuticle, which you can't quite see in this image because it's not being stained, is an epidermis. And then beneath the epidermis is usually a sclerified hypodermis that may be discontinuous or continuous, and may even be several layers depending on the taxon. And then what you see below that depends on whether you're in between a vascular bundle or above a vascular bundle. In between, you have the palisade mesophyll, where a lot of the photosynthesis takes place, and then the spongy mesophyll. And if you're above a vascular bundle, you see what's called in the literature a girder sclerenchyma, and really that's just a cluster of fibers above and also below the vascular bundle. And then the vascular bundle itself has the xylem and phloem, And then there's that other girder sclerenchyma and the abaxial epidermis. And there are a few other characters I saw as well that varied. The presence of these druce crystals, for one, and then also these fibers that would either be in the bundle sheath or scattered throughout the mesophyll. And I saw both sclerified fibers and cellulosic fibers. So that's an introduction to what a typical cross-section looks like. And so most species I saw had most of these characters in some form or another. So what did that variation look like? Well, I looked at 20 species, so I saw a lot of variation. And I'd just like to point out some key things on this relatively busy slide. So on the left here are just two images of the margins of leaflets. And you can see that the shape varies quite a bit. The presence of that girder sclerenchyma also varies. In this species on top, Oliviana, it's there, but in this one on the bottom, it's not. And also in this top one, the adaxial hypodermis is continuous, there's a single layer, and on the bottom, it has gaps. If you look at the center and look at this, these species with the red boxes, you'll notice that in some species, the girder sclerenchyma is completely absent in the middle of the leaflet. And in others, it's quite small, and in others, it's even bigger. And I tried to control for possible effects of age by always going to the center of the leaflet and always choosing mature leaves. And on the right here, this is just a reminder that there are a few species that are plicate, or those folded leaves. And that has its own effect on the morphology of the vasculature and the surrounding fibers. But remember, my question was not whether there is variation in this genus. My question is whether that variation is phylogenetically informative. So let's take a look at that. Well, here is a pared down version of my final matrix. I looked at a lot more characters than this, but there's a few. And the bottom line is that just like leaflet morphology, there's actually not very many phylogenetically informative characters. They tend to be spread across the phylogeny with no discernible pattern. And so these characters outlined in red here show no such pattern. However, I would like to talk about one particular character, and that's these cells that I'm informally calling flat tires because I actually don't know exactly what they are. I talked to anatomist Barry Tomlinson, and he's not sure what they are. There's a thought that they may have some secretory function because of the extensive plasmodesmatal connections between them and their neighboring cells. But the key thing for the purpose of my research is that they only show up in this clade down here, consisting of the Isthmus species, South American species, and this odd one, Zamia soconucensis. Initially, I thought it was diagnostic for this clade, actually, but it turns out they pop up a couple of times out of it. So I would like to look into this character more to determine their function and composition, and also to do some additional sectioning to see if they actually are only distributed in this clade, and to determine really what their ultimate distribution is. So where does this leave us? Is anatomical variation really very helpful in determining relationships between species in this genus? Well, there's definitely variation. It's there. But based on my initial survey, it looks like most of that variation is homoplacious and scattered across the phylogeny. However, there are a few characters, those quote-unquote flat tires included, that may be unique to certain clades. And it all begs the question of whether anatomy might actually be more dependent on ecology than phylogeny. In other words, it's possible that the variation I'm seeing has more to do with what sort of habitat these species live in than their phylogenetic relationships. And so that certainly begs another question and invites further research into whether there's an association between anatomical characters in this genus and the many different habitats they grow in. I'd like to wrap up with some final acknowledgements. You've heard many of the people who have been really instrumental to this project, but I'd like to send a final shout out to the Spec Lab, Shayla Salzman in particular, Dr. Barry Tomlinson, and a whole host of other people, including my funding sources, the Cals Alumni Association and the Dextra Undergraduate Research Endowment. And I want to thank all of you for tuning in this morning for the symposium. You have many more talks ahead of you. And thank you very much. I'd be happy to take questions.
Any questions for Rosie?
You don't want to be seen.
This is Marvin. I have a question. So if taxonomists haven't been able to use or haven't used anatomical traits to separate species and families, what traits do they use in the psychedelics to determine what's a genus and what's a family?
So there's some gross morphological traits that are helpful, such as the morphology of the reproductive structures. There are also some finer traits having to do with leaves as a whole, like the shapes of the leaves and the presence of spines on the leaf petiole. And then more recently, the vast majority of the phylogenetic work in Cycads is done with molecular phylogenies, including Michael Colontes.
Thanks, that was a really great presentation.
Thanks, Marvin.
Tom's got a question.
Tom?
So if these things aren't related to phylogeny, if they are consequences of the environment, what are the characteristics of the environment that give rise to these different anatomical features that you're seeing? What are some of the variations that could give rise to the presence or absence of the ribs or whatever you call those things?
Yeah, well, the genus has a very wide geographic distribution in many different habitats. And the one that pops immediately to mind when you ask that question is Zamia pseudoparasitica, which is actually the only epiphytic member of this genus. And it also happens to be the member that has the most of those unusual cells in it. And so as an epiphytic plant, the first thing that pops into mind for me is that it has to deal a lot with water stress. And so I don't know yet if there's an association between the presence of those cells and some improved capacity to contain water, but that's a potential avenue of inquiry.
I heard nothing. Okay. Great talk, Rosie. Great job. Okay.
Well done, Rosie.
Thanks, guys.
That's great. Our next speaker is Olivia Gomez, and she's concentrating in plant pathology, and she'll continue this interest pursuing a PhD next fall at Washington University in St. Louis in the Department of Plant and Microbiosciences. The title of her talk today is Biofilm in the Rhizosphere. Olivia?
Right. Hello. I'm here to present the research I've done last summer in the Donald Danforth Plant Science Center.
Okay, we're done.
So I'll start by introducing the root microbiome. It's essentially the collection of all the microbes associated with plant root. And this can be divided into the endosphere, the inside of the root, the rhizosphere, which is the soil immediately surrounding the root, and then the soil that is a little bit farther away. And the point is that a healthy microbiome leads to healthy plant root growth, longer and just more extensive. And this leads to drought resistance, which is of interest in cropping systems. And many bacteria in the wild form biofilm, which is a special growth habit that allows bacteria to live communally on a surface. It usually starts with a few individuals sticking, and then the colony grows, and as it grows, then it disperses, and the cycle repeats itself. In a previous study, Arabidopsis seedlings were grown in agar plates, and there were different treatments that were subjected to different mixtures of bacteria. Mixture A showed root growth comparable to that of the control, which had nothing. Mixture B, as you can see, has a shorter root phenotype than the control. But when you mix mixture A and B, you can see a rescuing of the short root phenotype. It's comparable to that of the control, if not even a little bit longer. And so what really intrigues us is what is causing this rescuing the phenotype. So the mixtures were sequenced, and the predominant genus of bacteria found in mixture A at the end of this treatment was Varivrax, while in mixture B, the predominant genus is Agrobacteria strains. And so the main question is, what accounts for these phenotypes? Why is it that there are different root lengths based on the different mixture of bacteria? And what is causing this rescuing of the short root phenotype when you mix the two mixtures together? The hypothesis is that biofilm production is what leads to the short root phenotype. In literature, you find that Varivrax, for the most part, doesn't form a biofilm, while Agrobacteria strains do. And the idea is that the Varivrax strains that will not form biofilm may disrupt agrobacteria biofilm production, and this might be what is causing the rescuing of the short root phenotype. So I was entrusted with 17 agrobacteria strains and 11 Varivrac strains. These were all from sorghum fields in the endosphere, rhizosphere, and surrounding soil, because it is hoped that this research will be applied to sorghum in the future. And so I measured the biofilm production of each individual strain, as well as combinations of different strains in vitro. So to summarize my findings, I found both biofilm lacking and biofilm producing agrobacterium. This is already contrary to the hypothesis. We only thought that there would be biofilm forming agrobacteria. Similarly, I found both biofilm producing as well as biofilm lacking Varivracs. We only thought that there would be biofilm lacking Varivracs. And so because we have these 4 different categories, here are all the different kinds of interactions I could observe. I made many different combinations and found some pretty surprising results. I categorized the different interactions as either synergistic, where they build each other up, disruptive, or neutral. And so one of the surprising interactions I found was that biofilm lacking agrobacteria can actually disrupt biofilm producing agrobacteria biofilm. Secondly, another surprising finding is that biofilm lacking agrobacteria can disrupt biofilm producing Varivracs. This is the actual complete opposite of what the hypothesis predicted. And thirdly, another surprising interaction is that biofilm producing Varivracs can actually have a synergistic effect with each other, that there's actually a greater amount of biofilm produced when you mix them together. Here are the different other interactions that I've observed, but they mostly fall into the predicted or predictable pattern. So in summary, biofilm production of Varivracs and agrobacteria is not so clear cut. There are some Varivracs that can produce or lack biofilm, and there's agrobacteria that can form or lack biofilm as well. Secondly, each genera has the potential to disrupt the other. So based on these data, I would not account biofilm to be responsible for the different root phenotypes that we've observed. And lastly, more studies are needed because this was all done in vitro. We don't know what really happens in vivo. I'd like to thank Dr. Ming-Cheng Chi, who was my postdoc advisor, Dr. Becky Bart, who welcomed me into her lab, all of the BART lab for all their help, and the coordinators, Donald Danforth Plant Science Center, and NSF for funding. Thank you.
Great job.
Good job, Olivia.
Okay, our next talk is from Christian Kanlian. And Christian is a plant science major with a minor in community food systems. He's also designed his own concentration in urban food systems. After graduation, Christian hopes to secure a job at Headwater Food Hub as a produce manager in Rochester. He'll keep us posted on that. And the title of his presentation is Reflections on Controlled Environment Agriculture. Christian?
Thanks, Mike. My name's Christian, and today I'm going to talk about some of my thoughts about growing food in cities in controlled environments. There we go. Okay. So I'm sure you guys have noticed that lots of humans are living in cities now, and that means they have really little connection with where their food comes from and how it gets to them, despite the fact that abundant research shows that kids, especially when you teach kids how food is grown, that they make much healthier decisions even throughout their entire life. And recently there's been huge venture capital investment on both coasts in this sort of technology and growing food in controlled environments. And so it's important to be able to read beyond the headlines a little bit and see if these companies are really living up and doing what they say they're doing. And so I spent 2 summers in New York City, the first working with middle and high school students at the Food and Finance High School and the Police Athletic League in Harlem. And so they were growing lettuce and herbs and vegetables that would be used in their culinary arts program, which was really cool. And then the following summer, I was at a small consulting firm in Brooklyn, Agritecture. And in addition to helping with their consulting work, I put together and distributed a survey with 3 other Cornell interns at different CEA companies across New York City. And we surveyed the whole spectrum of growing technology in the city, all the way from outdoor soil-based, all the way to indoor, fully supplemental light. And I want to stress that there's no real right or wrong amount of technology to use when you're growing food. It just depends on what you want to grow and for whom. And what we found is that people don't like answering 80-question long surveys that take an hour and a half. So we didn't get that many responses, but we asked some more qualitative answers. And so a lot of these insights are drawn from some anecdotes and some longer answers that people provided us. One of the strongest things we found is that people growing food in cities consider themselves as more than just growers. They see themselves as part of a community, sort of like I mentioned earlier, providing that connection with where food is grown and how it gets to people and all sorts of nutrition education associated with that. And then we also surveyed packaging from a lot of these companies. And what we found is that they really downplay the technology that is used in growing, even if they invested millions and millions of dollars in putting it together. And we sort of speculated that that's because people already have associations with words like local and organic and pesticide free. And so that's what they're looking for when they shop, rather than greenhouse grown, hydroponic grown, soilless, you know. So that was sort of interesting. And next time you're at the grocery store, I encourage you to take a look at that, especially if you ever see lettuce with the root still on, because that means it was grown hydroponically. And some of my lingering thoughts about this are, again, around what is being grown and for whom, the socioeconomic divide and accessibility of these crops. Those indoor facilities I mentioned in the first slide that have gotten all that investment, I focused on leafy greens and herbs, microgreens even, that have a really high turnover, but have to be priced at a really inaccessible point to be able to recoup the huge capital investment. So if we're talking about feeding people or feeding cities, that's realistically, that's not it. And then one of the other things is around labor, and 2 of the professors I worked on are working around curriculum to develop sort of 2-year programs that can get people into this workforce, because there's a real shortage of people doing this work. And I want to highlight 2 great organizations that I surveyed and met the owners of. Oko Farms in Brooklyn, which grows not just the vegetables pictured here, but also fish in an aquaculture system, which is great because it's more closed loop, less inputs, more sustainable. And then Sky Vegetables in the Bronx that hires formerly incarcerated people and people from the community that it's in. And so with that, I'd like to thank Neil Matson and Anu Rangarajan, the 2 professors I was supervised by, as well as Wythe Marshall and Maya Ezedine, the 2 grad students who helped us with the survey design, and Mason, Brian, and Kat, my fellow interns who are at those other CA companies. And I would love to take any questions.
Great job, Christian. Questions?
So this is Marvin again. What's the current thinking about so the energy inputs and outputs of some of these closed systems of indoor lighting?
It's a great question. A lot of these companies claim to be sustainable or, you know, sort of the future of food. But you could make an argument probably about water, but certainly not about energy inputs. And people even talk about doing like solar panels, but you need 5 acres of solar panels for every acre of indoor greenhouse and 25 acres for every acre of plant factories of stacked, completely artificial lighting. So it's certainly in terms of energy, it's not more sustainable.
Thanks.
Until we get fusion power.
Okay.
Other questions? Nice presentation. Great job.
Good job, Christian.
Okay, moving forward, our next talk is from Jennifer King. And Jennifer is a plants and human health concentrator. She's hoping to apply to graduate school and pursue her interest in botany. And the title of her presentation is Herbaria for the Modern Day Botanist.
So it's a collection of dried and pressed plant specimens. The first herbarium was started in the 16th century by an Italian botanist. Since then, it's grown considerably, And the largest collection is the National Museum of Natural History in Paris, France, and it has over 8 million specimens. However, recently there's been threats to the funding, which has had a lot of consolidation of collections. So a little bit about the Liberty Hyde Bailey Hortorium on campus. It was started in 1935 by Liberty Hyde Bailey. It later merged with other collections, such as the CU Wiegmann collection in the botany department. It's called a hortorium and not an herbarium because it has origins that started in horticulture specimens. Right now we have a little over 900,000 specimens, and we have strengths in the oaks, palms, and legumes. So I've had the opportunity to work with many different natural history collections associated with the Bailey Hortorium on campus. First, we have the paleobotany collection, where I helped organize and barcode the specimens. We have the plant anatomy collection, where I helped image the slides, and the conservatory, where I've helped create interactive QR codes, and then finally the herbarium, where I spent the bulk of my time databasing and imaging the specimens. And all of this work has gone to curation work, has gone to efforts to help to preserve these collections. So some may see herbarium as an outdated practice, but it actually holds a lot of modern day importance. There's been a strong effort to digitize and database these materials, as seen to the right over here. The top is databasing and the bottom is imaging. All these material can help further studies in identification and morphology, reveal patterns in climate change, a written record for nomenclature. They're a place for researchers to deposit voucher specimens, and also material for chemical and DNA analysis. So you may be wondering how you create a specimen. I'm going to walk you through the process of what I've done to turn plants into specimens, 2 steps at a time. First, you start off in the field collecting. As you can see from this picture right here, we're in a canoe collecting aquatic plants. So field collecting can take you to many different terrains, such as tops of mountains, in lakes, and in forests. When choosing a plant, it's best to find a specimen that displays roots, stems, leaves, flowers, and fruits. In addition, you also should be note-taking out in the field, writing down the locality, habitat, plant descriptors, and the date. Shortly after with your specimen, you will dry and press it over here as seen on the right. You will press it in newspaper, and you want one side of the bottom leaf and one side of the top leaf at least showing. You will dry until all the moisture is gone, usually about 1 to 2 weeks. So moving on, our next step is put your specimen in the freezer. This is a very important step, which is sometimes overlooked. It's important to put it in the freezer because that kills any pests or bugs that may be on the specimens that you don't want to get into the herbarium, which can eat the material. While your specimen's in the freezer, you can go ahead and create the label. You will do this with your notes and data that you took from the field. In addition, you can use a dichotomous key to officially properly key out and identify your specimen. From there, you're finally ready to mount your specimen on archival paper with glue and linen, and once that is dried, voila, you have your very own specimen that's ready for filing. So some takeaways that I've learned from my experience at the Bailey Hortorium. I've learned skills of curation, digitization, and methods in field collection. The Bailey Hortorium and other herbarium currently right now have strong pushes and focuses on digitizing their collections, all to create a virtual herbarium. And this is just an example of why herbaria still hold important value and that they're evolving with science and technology. My acknowledgements, I'd like to thank everyone at the Bailey Hyde Hortorium, in particular Kevin, Anna, and Peter. This concludes my presentation. Thank you for listening.
Great job, Jennifer. Any questions, folks?
We are a bit ahead of schedule, Mike, so.
Can I ask a question? This is Chelsea Specht.
Please, Chelsea.
So Jennifer, I was just wondering when you think about all the work you got to do with the herbarium, what was the most intriguing to you, and what might you think about pursuing as a graduate student?
Well, since my concentration is in medicinal botany, I'm really curious about like the chemical analysis you could do from the specimens, and kind of, I don't know, the ethnobotany side of that, and how they use the plants for medicinal purposes. I kind of want to focus on that part.
Great. Did you find any herbarium labels that had sort of ethnobotanical medicinal information?
Not directly on the label, but I've kind of helped create a little collection and digitized some ones that like I personally found that had been turned into like pharmaceutical drugs and stuff. So I did my own kind of own research and then found them out in the filing cabinets.
Great. Thank you.
Thank you.
Our next presentation is from Emma Kubinski. And Emma is a plant sciences minor. Her minor is in soil science, her major is plant science. Her concentration is ecology of natural landscapes. And the title of her talk today is Ecological Restoration at Ganondagan State Historic Site. Emma?
Okay, so this morning we'll be talking about the ecological restoration work I carried out at Ganondagan State Historic Site with New York State Parks Environmental Field Team for the past 5 years. We had a few overarching goals that guided our restoration work. First, we wanted to restore the historical landscape back to how it was when the Haudenosaunee inhabited the site in the 1600s. This provides both ecological opportunities and educational work. We also want to restore and maintain our ecological community's capacity for self-sufficiency. We want to identify short and long-term threats to prepare accordingly. We want to catalog our biodiversity to preserve what we already have. And we want to promote stewardship through education and outreach. To meet these goals, we do 3 main things: monitoring, invasive species control, and the actual restorative processes. So the first big part of monitoring that we do is vegetation surveys. So these vegetation surveys that we carry out are in our grasslands and paired with grassland bird surveys. These surveys allow us to understand how the changing diversity and structure of our grasslands as we restore them affect the threatened bird species. The other big aspect of our monitoring is invasive species mapping, where we identify the location and density of invasive species populations around the park. This takes up a big chunk of our summer, so it's really important to recognize how critical this is to our restoration work. So an example of this is on the right. This is one of our invasive species on site, James Rocket. And here's a map that our surveying produced. So as you can see, there's small populations sprinkled throughout the park. Because we were able to detect them so early, we can rapidly respond and remove these invasives before they could spread and become more of a problem. Here's another example. This is probably one of our most problematic invasives on site. This is house swallowwort. As you can see over here, this is another map. So in areas like this, where there's just kind of one little bunch of swallowwort, we want to do things like manual hand removal. But in places like this, where the population is still pretty widespread, we'd want to turn to things like chemical control. So shifting onto our control methods, as I mentioned, we do use chemical controls. Because of the landscape scale we're working with and just simply not having enough people or time, we do use herbicides. However, we do use hand control as much as we can and when it's needed. Probably one of our coolest control methods is we carry out prescribed burns. Prescribed burns get rid of invasives that are early sprouting in the spring and also stimulate our native warm season grasses. It was also used by the Haudenosaunee, so it does have educational purposes as well. So after we carry out these control methods, we have a pretty blank slate in terms of plant community. So one of the things the environmental field team does is we do native seed collection at our local parks and natural areas. Here I'm collecting Carex aurea or golden sedge, which is one of my favorite plants. We then take these plants and we propagate them out in greenhouses and then use them in our restoration plantings after we've managed the site. So with all this time and effort, a really important question to ask is it working? So here's a GIS maps I made over my time there. So the darker red is a more dense population. And as you can see, throughout the years, we've seen a significant decline in house swallowwort. And then to put another visual on it, here's a picture I took in 2016 of one of our grasslands. So all this plant right here, this purple, that's house swallowwort. And all these grasses are cool season non-natives. This is a picture I took right before I left this summer and headed to school. These big tufts of grass are a bunch of warm season native grasses, and there's not any pale swallowwort in sight. I want to thank the awesome people I worked with at the environmental field team and New York State Park and Ganondagan for all the amazing opportunities I was provided. Any questions?
Good job.
So this is Marvin. I have a question. So one of your goals is to reestablish the site similar to what the Haudenosaunee would have seen at the time. And it strikes me that there's a lot of open field at this site. So do you know how much was originally forest and how much was open field at the time it was sort of managed by the Haudenosaunee?
Yeah, so what we have to go off of for our restoration work was journal entries from the French. So they do describe a lot of grassland that was actually maintained by prescribed burning. There's actually there was a recent study published on where they think that there was a lot of these grasslands, and they are throughout central New York. So this is the location they think there would have been one. A lot of them were considered oak savannas. So as you can see, we do have trees in these grasslands, and a lot of these are oak trees. But they were maintained by the Senecas. They're not totally natural, I guess you could say, but that's the age we're trying to kind of restore it back to.
Okay, thanks.
Other questions?
Emma, of the control strategies you tried, chemical, burn, hand pulling, which do you feel like was most effective?
It definitely depended on the plant. I talked about every invasive plant we controlled. The list would go on for a while. So it really was dependent on the plant. Because like swallowwort was very hard, and because it was so widespread, it was probably the hardest to get rid of. I would say chemical was the most effective generally for invasives, just because we could work with such large areas.
Thanks. I don't have a very technical question, but do you ever foresee a time when this area will not have to be constantly monitored? Do you think it will actually become a more natural state eventually?
Yeah, so that is one of our big overarching goals, is that we don't want to have to maintain these sites forever. We really want to be able to restore the site's self-sufficiency, so that it doesn't constantly have to be managed. So we take a lot of steps. One of our big steps is that we do plant some native plants, and we pick them so that the plants that we're planting are gonna outlast. And because we're also working in such big areas, we're trying to kind of make almost buffer zones in these big areas around. So that is one of our big goals, because it is important. If it has to be interfered with all the time, then what's the point? So that is one of our big goals.
Okay, thank you.
Yeah, of course. Thank you.
Great. Okay.
Thanks, everyone.
Thank you. Beautiful. We have a lot of new guests. Okay, our next speaker is Alexander Liu. Alex has a dual concentration in plant pathology and plant computational biology. After graduating from Cornell, he will be attending graduate school at Washington University in St. Louis. And today he'll tell us about comparing drought response in 2 C4 grasses using transcriptomic analysis.
Yep. Hello, everyone. Today I'll be talking about the work I did at the Donald Danforth Plant Science Center in St. Louis this summer, comparing drought response in 2 C4 grasses using transcriptomic analysis. So the projected climate in 2080 is expected to have an increase in dry days and a decrease in soil moisture overall, and this will lead to increased instances of drought and longer instances of drought, leading to more drought-stressed plants, which could lead to more crop losses. And so the goal of this study was to identify drought-stress-related genes in C4 grasses. The two species of grasses we were looking at are Cyteria viridis, which is closely related to C4 crops such as sorghum, maize, and sugarcane, and is also a model organism. And sorghum bicolor, which is a drought-tolerant C4 grass and is an important food and biofuel crop. Both of these grasses have their genome sequenced and are relatively small and have a long and a short lifespan. They can be easy to study. So we studied these plants in a high throughput phenotyping chamber. The plants were automatically watered and grown within the growth chamber, and they would be taken out via conveyor belt to be imaged every day for further analysis. In addition, there is the high throughput phenotyping experiment, we planted the plants inside normal growth chambers, and after 7 days, we moved it to the phenotyper and grew them for another 2 weeks, where we began the drought treatment. And the drought treatment would be a water deficit condition where the plants are only watered to 40% of their soil water capacity. And we had a well water treatment, which is the control, where the plants were watered to 85% of soil water capacity. And then after we began the drought treatment, we took leaf samples roughly every 2 days until 2 weeks after the treatment begins. And for each species of grass, we planted 3 separate genotypes, each treated in the water deficit treatment and the well water treatment. And for each treatment, we had 4 replicates, each where we took leaf samples from 3 plants and combined them. And these leaf samples were then, the RNA was extracted from them and sent off for 3' RNA sequencing. And so all of that was done before I actually arrived at the Danforth Center in the summer. And so my project was to develop an RNA-seq analysis pipeline. And so the general flow of this pipeline would be to take the raw data, quality control and filter it for both like bad low quality reads and also low quality samples. And then take the data and do differential gene expression analysis on it. And then after that to compare differentially expressed genes within the species. And finally look for conserved gene expression patterns between sorghum and Cyteria. And so for our quality control filtering step, we did the principal component analysis, and this basically reduces the thousands of gene expression data and collapses it down to a 2D chart where we can kind of visualize it easily. And for the differential gene expression analysis, we used 2 packages, DSeq2 and EdgeR, which we would then compare to see which one was most ideal for our analysis. And so looking at the principal component analysis, we found that we could easily distinguish the 3 separate genotypes of sorghum here. And in Cyteria, we could distinguish, we could also distinguish the differing genotypes, although the A10 and TB1248 are clustered more or less together. The more interesting thing is you see these separate dots over here, which should correspond with the clustering, with their respective clusters down here, but they do not. And we think these are probably samples that were mixed up during the sampling process with the plants. So, or they were planted incorrectly at the very beginning. Since we're working with hundreds of plants at a time, mistakes happen. And this is why the quality control step is so important. But this also shows that the Soteria data is overall more noisy since the cluster is not as defined. And we also expected that the clusters would hopefully also appear when we compared drought-treated plants to non-drought treated plants, which was not the case. Something that was interesting was that if you look at if you labeled the samples by the date that they were taken, it appears that the younger samples would cluster together and then followed by middle-aged ones and followed by the older ones, showing that there's basically the samples would cluster more by developmental time. This was evident in both sorghum and Soteria. This is interesting because, especially in Soteria, the plants are relatively small and have very, very fast lifespan. They can flower in like 8 weeks. And so over the 2-week experiment, they would go from a 2-leaf seedling to flowering by the end. And so moving on to the differential gene expression analysis, we first focused on a global drought response across all genotypes. So we were looking for genes that were differentially expressed between all 3 varieties of sorghum and Soteria. We found that HR tended to find a lot more differentially expressed genes than DEC2. This is especially evident in Soteria, where DEC2 basically didn't find any differentially expressed genes until day 14. In sorghum, though, the numbers of differentially expressed genes using the found using the 2 packages are more similar, and there are a decent number of differentially expressed genes that are found by both packages. And so after looking at differential gene expression within species, we wanted to compare the differential gene expression between the 2 species. And so we pulled an ortholog list of orthologous genes between sorghum and Soteria off JGI. There were 26,000 orthologs in that spreadsheet. And we wanted to focus on the early drought response, which would be somewhere, we characterized it as somewhere around day 6, day 8 time point where there's the first peak is evident. And since DEC2 didn't find any differentially expressed genes in Soteria, we could only use EdgeR to do this comparison. So basically we took the genes that were differentially expressed in sorghum and Soteria and found the genes that are differentially expressed in both. And so there is a pretty evident peak at day 8, in this case of around 30 genes. And so in summary, the gene expression seems to be affected mostly by genotype and developmental age of the plant. When I was sampling plants for a later experiment, a more scaled up experiment, it seemed that these plants were not particularly drought stressed. They were, both treatments came out green and there's no leaf rolling. So a more extreme drought treatment is probably needed. There's a peak in differentially expressed genes at somewhere between 6 day and 8. And some future directions would be clustering and network analysis of the differentially expressed genes. And the lab is currently applying the pipeline to a larger phenotyping experiment with 94 varieties instead of 3. And we'll be using full transcript RNA sequencing instead of 3' RNA-seq. And the last thing we could do would be to combine the RNA-seq data with metabolomics data. And I would like to thank the Donald Danforth Plant Science Center and the NSFRU program for having me uh, allowing me to work at, intern there during the summer, and Dr. Alan Hubbard for mentoring me, and Dr. Ivan Baxter in his lab for having me there. Thank you.
Great job, Alex.
Nice job, Alex.
I have a question. So when you say you want to do more severe drought response, I mean, as a developmental biologist, we're often criticized because we do drought, we just don't water them, which is stupid. But what will you guys do? A longer time of water deprivation, or just a more extreme water deprivation? What are the plans there?
We were looking at more, severe water deficit. So watering them less.
Same amount of time?
Yeah, same amount of time.
And when you go to WashU, are you planning on rejoining Ivan's lab?
It's a possibility. I mean, I have to go through my rotations first, but yeah.
Hey, Alex, I have a question for you.
The computational side. So.
Alex, this is Tom Owens. I got a question for you.
Yep.
The two programs that you use to analyze the differential expression, DSeq2 and the EdgeR, it's not surprising that they give different results, but it seemed kind of surprising that they give such different results that varied between the two species. One was very consistent for one, and one was less consistent for the two. What does that tell you?
Well, I think it was more, the differences was more due to the RNA-seq data itself than the two programs. So we noticed that for the Soteria, like our first, like very initial quality control step was just do SNP calling on the RNA-seq data sets. And the clustering for this, you'd expect that the clustering for SNP calling would be pretty tight because you just, calling SNPs. If one variety has it, then it should have it, right? But when we were looking at the Soteria data, it was just really noisy. You didn't see any clustering at all, which kind of indicates that the data quality was not necessarily the best. The sorghum data was, we had a lot more data for the sorghum samples than the Soteria samples, so that could have been why.
So, but that kind of raises another question. How do you know that EdgeR is giving you the best data?
We don't really. Yeah, didn't really put any, final, like, I guess, decision on which one's best, mainly because we didn't really know. They both, one's probably, like DSeq2 seems to be more sensitive, less sensitive than EdgeR, but we don't really know which one's better.
So do people do sort of manual checking in different species to see how well these things work, or they're just programs that are out there that people use and don't really pay any attention to that?
I don't know. We didn't really we just basically followed what other people usually use. I mean, it seems to be used across different organisms. So.
I would say yes and yes, Tom.
Yeah, thanks. I think it's really important that people recognize the limitations associated with the commercial programs out there to do this type of analysis.
Yeah, for transcriptomic analysis, it's harder to do controls.
Yeah.
Any other questions?
Yeah, I have a question. This is Tim Setter here. It's actually related to Michael's earlier question. The lack of a very strong effect on your transcriptomics by the drought treatment. I'm wondering if your high throughput phenotyping data showed evidence of decreased transpiration rates due to stomatal closing, and also decreased growth. Height growth or a number of leaves, that kind of thing.
I personally don't really know. There was somebody else, there was a postdoc that was doing the analysis on image analysis on these data sets. And there was something. I don't remember what. I think it was like decreased leaf area for the drought stress plants compared to non-drought stress plants.
Mhm.
So
Well, those two things, stomatical closure and decreased expansion growth, are usually very sensitive to small decreases in water status. So those would be interesting to look at with the high throughput phenotyping data. The other thing is, I wasn't clear, did you sample the entire seedling or only the mature leaf portion?
it was only the mature leaves.
Okay, so not the expanding tissue at all. All right,
No.
that's important to know.
Yeah.
Good. Excellent presentation. Thank you.
Thank you.
I have one more question. Is that a picture of you in the back wall there at an earlier age?
I think that's my brother, actually.
Okay. Maybe it's a freshman photo.
We
Sorry.
look similar, so.
Great job. Okay.
Nice job, Alex.
Cool.
Thank you.
Our next talk will be from Claire Morrow. Her concentration is sustainable plant production. And after graduation, she hopes to continue to explore her love of plants and the natural world by pursuing a career in agricultural extension. The title of her talk today is Tarping for Weed Management in Reduced Tillage Systems. Claire.
All right, hello everybody. Can you hear me okay?
Yes.
All right, well, let me just start by saying that I hope everybody is healthy and in a place where they feel safe. Today I'm going to talk to you about the work I did at the Cornell Small Farms program, more specifically tarping for weed management and reduced tillage systems. So this was done in the Cornell Small Farms Reduced Tillage Project. That's a multi-year project. I believe it's in its 9th year right now. And that's important because when you adopt a reduced tillage system, you tend to see a yield gap at the beginning while the system adjusts. This was also an organic system, and it's particularly hard for organic farmers to adopt reduced tillage just because they tend to use it as a method of weed control since they don't have access to herbicides. So this project collects data on labor, yield, temperature, weed counts, pretty much anything you can think of, just so that we can really create a full picture for farmers of what it means to adopt a reduced tillage system. So when we talk about tarping, we're not talking about these typical blue tarps that you're probably used to. We're talking about these much larger black plastic tarps that are much thicker and require several people to move. And you apply those 2 to 4 weeks before planting and after harvest. In some cases, they're even overwintered on beds. And what they do is they block out the light and they heat up, hopefully killing perennial weeds and creating sort of a stale seed bed effect. So here I have a little video of us removing some tarps. You can see it takes a couple people to move, and we're revealing some nice, beautiful beds there to plant into. That was actually not a very fun day on the farm. We had some geese come through, and those tarps were absolutely covered in poop and not very fun to move. So I figured the easiest way to show our experimental design was just to show it to you. So here we have our planted acreage, and this was done at the Thompson Research Farm over in Freeville. And we have a randomized block set up here with our 4 blocks. And so within each row, we have a tillage treatment. There's a no-till, a shallow till, and a deep till treatment. And then within each row, we have mulch combination. So the gray represents compost, the white is nothing, and the yellow is straw. So then within each tillage, we also have tarps here. So there'd be a tarp on a no-till treatment, a tarp on a shallow till treatment, and a tarp on a deep till treatment here. So we have pretty much every combination of tarp, tillage, and mulch. So these are our results looking at weed counts halfway through the season. And you can see already that our tarp treatments have much lower weed counts than our non-tarp treatments, which is exactly what we would be hoping to see. And here are our results of weed biomass right before harvest. And you can see already that our tarp treatments have much, much lower weed biomass than our non-tarp treatments, which is exactly what we would be hoping to see. So just to show how this also reflected yield, I have circled the tarp treatments in red just to make it a little bit easier for you to see, and they're right next to their counterparts here. And you can see that in pretty much every category, our TARP treatments kind of have much higher, well, not much higher in some cases, but have higher yield than our non-TARP treatments. But where you can really see the difference in the field, it's very exciting. So here are two comparable systems. These are both no-till compost, and you can see our TARP lettuce bed here is just absolutely beautiful, and our non-TARP one was absolutely covered in chickweed and all of these things. So I would just like to acknowledge Ryan Maher, who is the project lead and was very fun to work with over the summer, and the program director, Anu Rangarajan. And I had a wonderful summer there at the Small Farms program. Are there any questions?
Great job. Any questions from the crowd?
Hey, Claire, this is Tony. I was wondering if you think using TARP, it seems like it's limited to maybe smaller farm operations. Can you see this kind of expanding to larger operations, or are you limited by just having, you know, the size of the TARP? What are your thoughts?
It definitely, that is a limiting factor, just because it's so labor intensive to add and remove the tarps, and as you make the tarps bigger, it becomes even more labor intensive. You can get larger tarps. In terms of if a larger farm could adopt it, I feel like this really does have some potential if they're willing to put in the labor effort required, just because of how we saw, you know, such fewer weeds. So then it also reduces, you know, weed cultivation time that we'd have to do, you know, during the season and stuff. It was a lot of labor and it was hard, so it is limiting, but, you know, I think it has potential just because we got such great data, you know, that was really good results.
Great, thanks, Claire. No, that was great. Thanks for the presentation.
Hi, Claire, are those tarps reusable? And if so, like how many times could they be reused?
Yeah, they are reusable. Some of them are a little beat up, but we've had tarps that have lasted the entire project pretty much, which is cool to see. And that's 9 or 10 years, which is great. But yeah, as long as they're stored properly and you're careful when you're taking them on and off, you can reuse them, which is great to see, which is a lot better than the black plastic you typically see on an organic farm that just gets thrown away.
Yeah, great. Thank you.
Hi, Claire, this is Neil. I was just curious how long the carryover effects of the TARP might last. So, you know, one could envision maybe the first month or two, you have really good control, but by the end of a growing season, maybe there would be less control.
Yeah, exactly. And that is kind of what we saw, was that, you know, over the course of the growing season, there were a lot more annual weeds popping up, but this did do a great job controlling our perennial weeds. And I guess what it was mainly used for was to just we got a really good jump on the weeds instead of at the very beginning of our season having to deal with it. We had some time where plants were getting established and they were really free of them. And the effects tend to, you know, diminish over time, but it gave us a really good jump on the weeds. And you can see it reflected in the yields too, that our tarps really were helping our plants grow and get established well, which was nice to see.
Great,
Claire,
thank you.
Claire, are there data on tarping in multiple years? So if you tarp the same field 1 year, the second year, the third year, do you see a reduction in weed populations and weed pressure over time?
Yeah, so I gave you data on the year that I worked when we did the lettuce crop, right. But this data does look a lot better than the first year that they did tarps. And over time, yeah, it does get less and less over the years with the reduced tillage system, which is really cool to see.
There's a question from John Forrest. It says, were the tarps reused?
Yep, these were just rolled up and stored by us after they were removed and then put back on by us afterwards.
Great, thanks for that question, John. Other questions for Claire?
What did you guys do with the harvest?
Well, we donated what we could to the Southern Tier Food Banquet, and we also had Dillman Hill take some, which was nice. So maybe if you had a Dillman Hill CSA, you got to have a little bit of our lettuce. Unfortunately, a lot of it, just the field heat as we were harvesting, got a lot of the lettuce looking not so great. But we donated what we could, and we had a whole big box truck be taken, but unfortunately a lot of it had to be composted, which was not fun to see.
Okay, great job.
Nice job, Claire. Excellent shout out to Dillman Hill CSA.
Okay, next up we have Peter J. Oppenheimer, and Peter is a plant pathology concentrator. He's also got a second major in environmental and sustainability sciences. After graduation, Peter will pursue a PhD in plant pathology at North Carolina State University. And he's going to study Fusarium mycotoxins in wheat. And today his title is Using Mass Spectrometry to Analyze the Host Response in Nicotiana Benthamiana to superinfection by 2 unrelated viruses. Hopefully he will explain that better than I just announced it.
Thank you, Mike. Can everybody hear me and see my screen okay?
Yep.
Yep.
Awesome. Yeah, thank you everyone for being here. Today I'll be talking about the research I conducted last summer at the National University of Singapore, where I used mass spectrometry to analyze the host response in tobacco to superinfection by 2 unrelated viruses. So the phenomenon that we were studying in our research is called heterologous superinfection exclusion. It is analogous in some way to a vaccine for a plant, except of course the mechanism works very differently. So superinfection exclusion is the ability of an already established virus in a plant to prevent the subsequent reinfection of another virus. And when these 2 viruses are distantly or unrelated viruses, it is known as heterologous superinfection exclusion, or HSE. And in our experimental system, our two viruses are TMV43A and cucumber mosaic virus. TMV43A is a mutated virus of TMV, which replicates much slower and also produces few to little symptoms in the plant, and it is protecting infection from CMV, which is a very more virulent virus. It causes symptoms of severe mosaic and systemic necrosis and has the widest known host range of any plant virus. And so the question that we were asking in our research is, how does superinfection with this mutant strain virus affect the CMV viral accumulation in tobacco? And we predict that viruses that are superinfected will see a decrease in CMV accumulation. Here is our experimental design. As you see, groups number 4 and number 5 are our superinfected plants. And we also have plants that are singly infected with CMV and TMV43A. And we are collecting RNA and protein samples on days 4, 10, and 16. And we are comparing these RNA and protein samples from our superinfected plants to our singly infected plants. So when we extract our RNA, we will use that to amplify the viral coat protein in the virus to confirm that the plants were correctly inoculated and infected with the correct virus. The proteins will be used for both a Western blot, which will also amplify the coat, will also show the coat protein, but it will also show us the quality of the protein itself, which is important because it's important for mass spectrometry, which is what will give us differentially abundant proteins. And so we can compare the abundance of certain proteins in our superinfected plants to the abundance of proteins in our singly infected plants. And from the differential regulation of these proteins, we can come up with possible mechanisms of why our cross-protected plants are not seeing fewer CMV, lower CMV accumulation than singly infected plants. And so our results showed that CMV accumulation decreased in superinfected plants, which is positive. And it also showed that proteins associated with photosynthesis were significantly upregulated in superinfected plants, and proteins associated with fatty acid synthesis were significantly decreased in superinfected plants. And so we believe this is significant because CMV often attacks the photosynthetic pathways in tobacco. And so by increasing regulation of photosynthetic pigments in superinfected plants, we believe it mitigates some of the damage done to the photosystems and allows the tobacco to have the necessary energy to defend against the infection. And we also, interestingly, saw the decrease in fatty acid synthesis-related proteins, which we believe inhibits the formation of the VRC, viral replication complex. So CMV is a virus that requires a lipid envelope to be constructed before entering a new cell. And this viral envelope is composed of lipids. And these lipids, it accrues from the host, the host's machinery. And if the host downregulates the synthesis of lipids, the virus will be starved of lipids and will be unable to form this lipid envelope and thus be stuck in the cell and will replicate much slower. And so the implications of this study are wide-ranging because to our knowledge, this is the very first report indicating that a virus is able to exclude infection by a completely unrelated virus. And this report can go a long way to inform future research of the possible mechanisms behind HSE. And maybe one day we will be able to create, use this for biotechnology to create a protectorant preventing CMV infections in crops. I would like to thank the members of my lab at the National University of Singapore, Prem, Yang, Mercy, and Dr. Sekwan Wong for being very gracious towards me and teaching me all the necessary lab protocol and for my research. And I'd also like to thank the CALS Global Fellows Program for providing me with this opportunity and help me become accustomed to life abroad in Singapore. Thank you, and does anybody have any questions? I can answer them now. Thank you.
Good job. Questions for Peter?
Peter, that was a really exciting piece of research. Do you know, does the mutated tobacco mosaic virus provide some resistance to the wild type of tobacco mosaic virus?
So this experiment was preceded by another experiment in the same lab where they used wild type TMV as the challenger virus and also found that TMV 43A was able to cause a decrease in TMV viral load. And I forget the mechanism behind it. It had to do with ROS species accumulation. I know that.
Very exciting.
Yeah, thank you.
Peter, do you know if this would work in a plant beyond Benthamiana?
Have I worked in a plant behind,
would this system, have they tried it in tomato or in a cucurbit?
I don't know. I don't know if they've tried it in other plants. I'm sorry, I can't answer that question. I know that the other experiment with TMV 43A was also done using the same host. I think I also remember seeing research where they created this mutant strain virus in a different experiment. And I think I remember that being done on tobacco too, but I'm not entirely sure. Thank you. Good question, though.
Cool, thanks.
I see nothing on chat. Okay, great job, Peter.
Thank you.
Next up, we have Cassandra Proctor. And Cassandra is a concentrator in plant molecular and cellular and developmental biology, the physiology concentration. Her plans are to take a gap year before entering graduate school. And the title of today's talk is Investigating the Role of Replication Factor C, or RFC, during arbuscular mycorrhizal symbiosis.
All right. So as Mike said, I'm going to be giving my talk on the project that I've been working on for my honors thesis in the Harrison lab, titled Investigating the Role of Replication Factor C genes during the arbuscular mycorrhizal symbiosis. hang on, my screen is blocked on my face. Anyway, as an introduction, so the arbuscular mycorrhizal symbiosis is an endosymbiotic association between a family of fungi and the roots of many land plants. And in exchange for different mineral nutrients that the fungus provides the plant provides the fungus with a carbon source. So what happens is the fungal spores lay dormant in the soil until a plant root grows close enough where the spore will start producing these hyphae that will grow either out into the soil or towards the plant root. Once they reach the plant root, they form this specialized structure called a hyphopodium, which allows the hyphae to penetrate the epidermal cells and grow into the inner cortical cells, where they form these highly branched structures called arbuscules. And this is where this is the site of nutrient exchange between the fungus and the plant. And interestingly, this symbiosis is very ancient and it's highly conserved in plant species. It's believed to only have evolved once in the first land plant, and plant species nowadays that can't form the symbiosis have lost that ability over time. So, based on this highly conserved ability, a phylogenomic study was done that compared the genomes of 50 different plant species, 11 of which had lost their ability to form the symbiosis. And the idea was that you could get a list of genes that were hypothesized to be conserved for the AM symbiosis, because genes that were only found in the AM capable species probably, and not the species that were unable to form the symbiosis, were probably conserved because of their ability to function in the symbiosis. And this study yielded around over 100 orthologs in the model species Metacoga truncatula, which is the species that's been used in the study. And 2 of these genes found in Metacoga were annotated as replication factor C genes, which we named RFCA and RFCB. And interestingly, in this tree with the other RFC proteins, you can see there's 5 different plant species, and Arabidopsis is the only one in this tree that has lost their ability to form the symbiosis. And in the RFCA and RFCB clades, you see Arabidopsis is missing. So more about the replication factor C complex. This complex is essential for DNA replication and repair in all eukaryotes. And what happens is the subunits come together to form the complex, and they bind pCNA, or the proliferating cell nuclear antigen, which is a clamp loading protein. And so when the subunits bind the pCNA, they open and close it around the DNA, they release it, and then the pCNA can recruit the polymerases needed for DNA replication to occur. And while this complex, like I said, is essential for all eukaryotes, the arrangement of the subunits varies across species, and it's not known in Metacoga truncatula. So moving on to the main objective of this project, it was to functionally characterize RFCA and RFCB genes during arbuscular mycorrhizal symbiosis by first, determining the spatial expression patterns of the genes, second, studying the AM phenotype of a double mutant, and 3, analyzing the structure and sequence of the proteins RFCA and RFCB to identify potential functions. So going to the first objective, promoter Gus constructs for RFCA and RFCB were transiently transformed into the roots of Metacoga truncatula. And as you can see here, in blue is shown the Gus expression. In green, you see the WGA expression, which is a fluorescent protein that stains fungal cell walls. So this green here will show where the fungus is in the cell. And for both RFCA and RFCB, you can see the Gus and the WGA overlap in the merged images, which means that these proteins are being expressed in cells that contain the fungus, so cells containing arbuscules, which is interesting because this supports what was found in the phylogenomic study, that these genes are most likely conserved because they function in the symbiosis. And because they're found in cells participating in the symbiosis, you can see that that supports the hypothesis. Moving on to the second objective of the project by studying the AM phenotype. So in the phylogenomic study in 2016, it was also published that an RFCB mutant showed an overall reduction in root length colonization at 5 weeks post-planting. And the root length colonization is just the overall amount of internal hyphae in the roots. However, when I looked at the double mutant, there was no change in root length colonization found. But visual analysis of the visual analysis of the arbuscules actually showed that there might be a potential mutant phenotype, because as you can see in the wild type, the arbuscules look very healthy. They appear to fill most of the cell. They're highly branched. Whereas in the double mutant, you see some almost wild type looking arbuscules. You also see a lot more that just don't appear to be as healthy or degenerative. So when this was analyzed, actually, there was significant decreases found in both the arbuscule area and the proportion of the cell filled by the arbuscule for the double mutant in both the 3 weeks and the 5-week post-planting samples. So then moving on to the final objective, a gene sequence and structure analysis of RFCA and RFCB showed that they might serve different functions. So what happened was, as you can see here, all of the RFCAs have this conserved, RFC proteins have this conserved RFC domain, which is the reason that RFCA and RFCB were annotated as replication factors. But in addition to these domains, both RFCA and RFCB have these unknown domains that are about the same size as the RFC domain. So the idea here was a blast search was done with the amino acid sequence of the unknown domains to see if any sort of hits would give us an idea of where these domains might have come from. However, the results of the blast search after they were aligned and maybe the tree only produced other AM-specific RFC genes. And interestingly, the RFCA results only showed other RFCA proteins in other species, and the RFCB only yielded RFCB results in other species, which gives the idea that these domains might not be homologous. So moving on to some discussion points. First, as I mentioned before, the gene expression data really supports the hypothesis from the 2016 phylogenomic study that the RFCs were conserved due to their function during the symbiosis. Additionally, the compromised arbuscule development in the double mutant suggests that one or both of the genes are necessary for the healthy symbiosis. And single mutants are really need to be analyzed to look at arbuscule development to see whether it's both of the genes causing this compromised development or if it's just one. Finally, the different unknown domains in the genes may suggest that the two have different functions, which again highlights the need to analyze the single mutant plants to determine really if they do play a similar function or if they're doing two different things during the symbiosis. Finally, I'd like to acknowledge my PI, Dr. Maria Harrison, and my postdoc mentor, Dr. Armando Bravo, for all the help and the opportunity that they gave me during this project, as well as the rest of the Harrison Lab for just the friendship and the mentorship that they've offered me for the past 5 years that I've interned and worked in the lab. And I finally like to acknowledge just that this project was funded by the American Society of Plant Biologists Summer Undergraduate Research Fellowship and the NSF Plant Genome Research Grant. Thank you, and does anyone have any questions?
Good job.
Nice job.
Cassie, I got a question related to the domains on the two proteins that you said only map to A genes or B genes. Have you looked any more at the structure of those genes? I mean, you can do a lot of structural analysis on the sequences that should tell you, are there similar domains? Are there different structural domains, even though they may not match up to other things? Have you looked at that at all?
I've tried, I've did different types of blast search, like PSI blast search that went back and back to see if there was anything that could come up as anything other than the replication factor. I've also used some different programs that tried to figure out different structural parts of the domains, like looking for nuclear localization signals and all sorts of other stuff. And I really wasn't able to find anything conclusive. As of right now, I do not know where these domains are coming from, and I don't have an idea of what could be doing. But there is evidence that either they come from two different places and they're not homologous, or they just diverged so long ago that you can't find the similarity needed to find the homology criteria.
Great, thank you.
Okay, beautiful job. Thank you.
Great job, Cassie. Yeah, nice job, Cassie.
Okay, our next presentation is from Benjamin Rouse. Benjamin designed his own concentration in physiology and ecology of crop plants. After graduation, he will enter industry while establishing a farmstead breeding program for colored raspberry and blackberry. Today's talk is understanding how inherent noise measurements place limits on high throughput phenotyping.
For those of you who do and do not know me, my name is Benjamin Rouse, and Dr. Tom Owens helped mentor me through this research of understanding how inherent noise measurements place limits on high throughput phenotyping. So going over a little bit of the roadmap for the talk here, high throughput phenotype measurements are based on the direct relationship between fluorescence and photosynthesis, and we're going to talk about some terms that need to be defined in relation to fluorescence, and they will help us derive a light induction curve. And then we're going to help us understand the instrument noise contribution, and then our strategy to overcome this problem will then be shown. So a little background information. Here's a picture of what we know as light energy will enter a plant, and there are competing processes for this energy within the plant against electron transport found in photosynthesis, and fluorescence is one of those competing processes. Fluorescence can simply be defined as a readmission of a photon. Our picture here shows chlorophyll dissolved in alcohol under white light, and it's fluorescing green, as opposed to it still being dissolved under alcohol, but UV light shows that it's red. So these FO and FM signals here will be read by the high throughput fluorometer, excuse me. And since we know that fluorescence and photosynthesis are directly related, getting an accurate measurement of the fluorescent signal is important in order to get an accurate photosynthetic phenotype. So these signals read by the machine are understood as light induction curves, and due to time, even going through a generic light induction curve would be impossible. But I would like to introduce the fact that the chemistry and physics gets much more interesting really quickly. The big takeaway from this slide is that all the measurements associated with the photosynthetic rate are scaled to the dark adapted FO value. To simplify things a bit, we're going to start by dark adapting a sample. We're going to take a sample, we're going to put it into a dark room, and by doing this, it allows the chlorophyll to have the maximum capacity to use its light energy, driving electron transport. We apply a modulating light to our sample. It's a very low light intensity that does induce some fluorescence, but it doesn't change the sample's photosynthetic physiology. Put another way, with the modulated light on, the sample remains dark adapted. And because we know the capacity for photosynthesis in a dark adapted sample is maximal, the fluorescence level is minimal. We therefore call this level FO. To reveal the other extreme of photosynthesis, we need to expose the sample to a short pulse of light that saturates photosynthesis. Therefore, we call the maximum level of fluorescence FM. Although the absolute amount of fluorescence from any leaf is highly variable, the ratio of FM over FO is highly conserved at about 4.92. For this reason, the values of FO and FM are used to normalize all fluorescence measurements so that those measurements can be used to make comparison between leaves or treatments. Considering the time scale we're concerned with, the noise is not of biological origin. The noise is due to the instrument. In fact, noise is present in all instruments at some level. The contribution of the biology to this problem is the variation in FO between different samples, meaning many different FOs. If the noise level is due to the instrument, but biology causes FO to vary between samples, then the signal to noise ratio gets smaller as the level of FO fluorescence gets smaller. What this means is that when FO levels are low, such as below 0 values, determining the actual value of FO is not possible, and the data from that sample cannot be used. Our FO array helps us show the variation of FO during high throughput application. The F intensity very clearly increases into the upper right-hand corner direction here. Remember now, FO is used to normalize all these subsequent and light adapted measurements for photosynthetic phenotyping. The experimental design is then to make FO artificially small. By doing so, it allows us to see when the error occurs. To do this, we look at 3 different maize samples with at least 3 different areas of interest. The picture seen here is one of the maize leaves, and each with 6 different areas of interest, each with a different FO value. Now, so the neutral density filters were placed between the sample and the detector. And it should be noted that the neutral density filters only change the intensity of the fluorescence signal perceived by the fluorometer and not the light conditions that illuminates the sample and establish its physiology. Thus, we're able to isolate the noise found in the machine. So we understand that the inherent instrument noise contribute to the high throughput measurements of photosynthesis. We know that they arise as a consequence of the natural variability in FO fluorescence levels of the leaves, and we know that the samples with FO values that are low compared to the average FO levels in an array cannot be measured. There are two possible ways to overcome this: sorting samples by FO levels before measurements, which diminishes the inherent high throughput character, or get manufacturer to modify a code to allow greater averages for FO and FS measurements, since they're steady state conditions. Lastly, and certainly not leastly, I want to not only just personally acknowledge Dr. Tom Owens, but I wanted to extend my deepest respect for his integrity and care that he's shown working in this project. His instruction, guidance, and patience will continue to teach me the rest of my life, and thanks, Tom. So I'll be happy to take your questions.
Fabulous.
Well done, Benjamin.
Questions? Tom is smiling big. I see that.
That's what we're supposed to do, right?
Yes, yes. Sorry. We're all doing that. No questions? Okay. Here we have Raul Campo Lizama, and his concentration is in plants and human health. And his title is Grandma's Potions, Discussions on Herbal Remedies and Cultural Identity Among Latinx Individuals.
Okay, so hi everyone. My name is Raul, and today I'm going to discuss research that I did in the fall of 2019. So I'm really interested in ethnobotany, and more importantly in the ethnography, because I've always felt like that part has been kind of not discussed in like ethnopharmacological or ethnobotanical research, and it's actually led to some pretty vile things, you know, like biopiracy and things like that. Additionally, I'm interested in the way that ethnomedicine and migration kind of interact with each other. And in the summer of 2019, I had the opportunity to do an independent research project where I arrived to this research question, right? So how does ethnomedicine influence the cultural identity among college-age Latinx immigrants, right? So basically what I did is I interviewed people and asked them about their use of ethnomedicine and how that interacts with their cultural identity. So because this was a quick pilot study, I had no official hypotheses, but I did have my opinions informed by the literature. So this is basically what I did, right? The study lasted about 3 months. I interviewed a total of 47 people in both English, Spanish, and some Mayans. And the methods that I used was ethnography with informal, semi-structured interviews. I gather ethnobotanical data and ethnographic data, which I'll show you the ethnobotanical data right here. And as you can see with the ethnobotanical data, you have everything from family, starting with, then you have specimen, the popular and the parts used, the preparations, the applications, the percent cited, the elements it cures for, and the pharmacological activities at the rightmost side. And yeah, so how does all of this lead back to my question, right? How does ethnomedicine influence cultural identity among college-age Latinx immigrants, right? In my ethnography, I learned some essential things, right? And that is that the use of ethnomedicine can help to deal with the trauma of migration and other downstream social effects like racism and xenophobia. And mind you, this is all a pilot study, so this is something that needs to be like for further addressed and research. But essentially, in my ethnography, I was able to find these patterns, and I'm about to show you. But I also want you to know that I'm shortening this a lot because of time. But these things are much more nuanced than they appear. So we have something like migration and cultural identity, which are things that can actually affect people's self-esteem and self-value. Additionally, identity is highly mutable, especially in the light of migration. It is affected by social positioning more than we like to accept. Another thing is that when people from marginalized communities migrate to a country that's hostile towards them, they experience something called cultural bereavement, which meaning people mourn the loss of their culture, and this usually comes from the pressure of forced assimilation, which can lead to a variety of mood disorders like anxiety, depression, and in fact, migrant populations have the highest indexes of untreated mental health issues. Though something that can help with these issues is this idea of performing culture, or like feeling like one is connected to their roots and things like that. And particularly what helps is food and religiosity. And something that's at the apex of food and religiosity is ethnomedicine. So according to many studies, performing these parts of culture can help with can help with the effects of acculturation and more successfully integrate immigrant societies into the current into the current culture. There's also a very interesting thing about ethnomedicine that used to its like mystical component. It's very unique to the culture that it comes from. This ultimately connects people with their loved ones, and it makes much more sense in the light of ethnomedicine of why certain like clinical perceptions of ethnomedicine is perceived as opposed to Western biomedicine. So why is all of this important, right? So so in a more conceptual, like big picture thing, is that 80% of the world uses herbal medicine as primary healthcare. And this is crucial to addressing global health issues and so forth. But things like this can also help with pharmacological research through various inverse methods of pharmacology. Finally, it's very important to detail the clinical experiences of marginalized communities to provide a more inclusive and more competent care. So I want to give a talk to the Cornell McNair Scholars Program, who really helped me out and have always been like such a pillar of strength and hope for me, and to my mentors, Julia Frizo and Rachel Dudley. And if you have any questions, please let me know. Thank you so much.
Good job, Ro.
Thank you.
Questions? Neil?
Raul, this is Neil. Very nice job. I was curious in the interviews, how did people address so taking herbal medicine in the context of not being, you know, in their culture with their family members? So would they still glean benefit, but was it not as much benefit because they weren't there with their grandma or family members? So kind of more related to that context.
So what I saw in my ethnography is that a lot of the parts, when people mentioned that they were using ethnomedicine, it was usually because they were like, it usually comes from somewhere. That's like the like one of the main things of ethnomedicine. It's passed down like intergenerationally, right? So a lot of people were saying that like it was less effective because it was something they were have to administer themselves. And a lot of parts also in the literature, ethnomedicine is it seems to be much more like effective when it's administered by like a community member or someone that is perceived to be very knowledgeable in these in this, which is really interesting. Yeah. But there's also some studies that like show like the increased pharmacological activity or placebo activity of certain like ethnomedicinal treatments, as opposed to like controls and stuff like that. But yeah.
Thank you. That's great. Yeah, that really addresses my question.
Did you enjoy doing all the interviews, Raul?
Oh my gosh, yeah. So it was essentially everyone, like it was, a lot of these people were my friends. I did have to discard some interviews because of that, but it was everyone from like my community here at Cornell. So it was like a very like personal project for me. But yeah.
All right. We are.
Okay. Thanks, Raul.
Thank you.
Our next talk is from Samuel Sternbach, and Samuel is a concentrator in soil science. His talk today is the effects of herbicide on loblolly silviculture.
Okay. Hello, everyone. My name is Samuel Sternbeck, and over the previous summer, I worked at North Carolina State University for the Soil Science RE. And there I worked with Rachel Cook, who is a professor of silviculture. So silviculture is the processing of trees for lumber or pulpwood. Specifically, I was focusing on loblolly pines, which, as you can see, relatively tall trees, and the pine cones we say are about the size of a potato, tater, Pinus taida. Easy way to remember it. So common in the southeastern United States, particularly North Carolina, which is where we were. And then the two most common ways to prevent weeds from popping up in this system is using imazapyr or sulfametoran. Imazapyr is used for herbaceous weeds, while sulfametoran is used for woody brushes. The problem is that in silvicultural systems, it's very hard to get weed cover reduced, because as it is a pine forest, there's not much time between seasons where you can spray it down just through the air. As you can see, the two main ways are the helicopter overhead flying or just going in manually and spraying. But as I said, as it's a pine system, you can't spray it after it has the canopy cover, and then the manual walking around is labor intensive. So in this experiment, we were trying to figure out what is another way to reduce the cover of woody and herbaceous weeds. So the first way we were thinking is the hardwood release system, which is spraying the herbicides after the trees are established and planted. And then the other method was the site preparation method, which is spraying the ground and covered before the trees are planted and then after the trees are planted. And then just a side note was that the soil that we were looking at was a Little Joe series, which was fine mixed active, but the most important part was that it is a well draining soil. So the purpose of this, as I said, was to quantify the relationship between the densities of the hardwood pines and then the weeds, and then to also determine the effects of the different timing to see which one was more effective and if either of them had a larger benefit. So to quantify how effective each system was, we checked the heights of the trees, the live crown, species covering, and also the DBH. DBH was measured using this tape you can see on the bottom, which was specifically a DBH tape, and you would wrap it around the tree, and that would give you the diameter rest height. And then on the inclinometer, you step a specific distance from the tree, and you would measure it from the bottom to the top, and that would give you the height. So these are the results. On the left, you can see the untreated, where there was no spraying, and you can see that it's very covered, and it's even hard to see through the undergrowth to the pines. The site release was slightly better, but you can still see you can see the pine trees, but there's still some cover compared to the site prep, where there's barely any cover, and for the most part, there's all pine trees. So this was the actual quantitative data. For the untreated and the site prep, there was the greatest significant difference, but compared to the untreated and 2-year release, there was not a significant difference. So we can say that there was a significant difference between the untreated and site prep, which was when there was a spraying of the herbicides before the trees were planted. And then you can see that there is a slightly significant association between the pine basal area and the hardwood basal area, so there is an improvement and a need to continue working on decreasing the hardwood and increasing the pine basal area. So I'd like to thank everyone at North Carolina State University that helped with this project, Rachel Cook and her lab technician manager, Morgan Schultes, along with Jerry Creighton, who was person in charge of the forest that we were using for the experiment, and then also North Carolina State University and the RAU. Thank you, and I'd be happy to answer any questions.
Great job, Samuel.
Hi, Sam. This is Marvin. I can understand the need to have sort of these monocultures of pine trees there for wood production, but has there been any thought about increasing the diversity, at least after establishment, to maybe mitigate some of the pest problems that might occur in a heavily monocultured system?
Yes, there is. However, it's at this point still most effective to go with the monocultures just because of the mass production and the amount of bulk wood that you're able to pull in, because it's only over a 20-year period that you need to keep it safe for, that you could just, after the 20 years, plow it over and plant a different monoculture. So unfortunately, monocultures seem to be the way to go for now.
Okay.
Yeah, one of the things that I found impressive about this was that it was a 20-year experiment. That's some serious forethought.
Okay, great job, Samuel. Thank you. Our next talk is from David John. His concentration is plant pathology and plant microbiology. And his talk today is entitled Developmental Changes of Carbohydrates in Peach Flesh.
Thank you, Mike. Can you hear me?
I can.
Thanks. And hello, everyone. I'm David, and today I'd like to introduce you my experimental learning project, Developmental Changes of Carbohydrates in Peach Flesh. In this winter break, I worked in Dr. Lai Liang Chen's lab, and the first question here is, why are we doing this? Why we tested the carbohydrate level in peach? Here are two reasons. The first one is carbohydrate level is directly related to food quality and flavor, and another reason is we want to base on this project to find out the developmental regulation of synthesis of secondary metabolites. And here's the materials. 147 samples were collected. They're from 6 cultivars. There should have 7, but actually 1 cultivar was infected by black rot, which is a sad story. And to show the developmental changes, we collect them in 9 days, and the time span is 3 months from April to August. In each group, we have 3 or 5 repetitions. On the right, you can see there are some colorful pictures, and each color actually represents 1 cultivar. Here's a larger picture. As you can see, oh, sorry. As you can see here, this row represents the cultivars, and this is the collection date, and here's a dry weight and some dates like that. All samples were processed into the freeze-dried ponder because they are much easier to handle with. The first step of my experiment is the extraction. We put this ponder, we add the methanol and the ribitol into this ponder and centrifuge a few times, and a little bit ponder on the bottom of this well is sugar acids, the carbohydrates we need. And the next step is the derivation. The chemical formula looks very complex, but the main point here is we want to make the polar molecules like sugar acids more volatile. We want them more volatile because the next step is the GCMS. From the name, you can tell that this technology is the combination between the gas chromatography and the mass spectrometry. The advantage of GC is it's really good at separating different compounds from a mixture, and the mass spectrometry can accurately detect what the molecule is. So in this project, the sample was injected here and went into this column. They are separated here, and the detector will tell us the concentration and what the molecule is. In the right, you can see a GCMX machine in Dr. Chen's lab. It was working at the time I took the picture. The whole project was still unfinished, and so I'm so sorry that I cannot present some conclusions or takeaways, but here is raw data from the GCMS. This is based on 1 sample. Here you can see many peaks, and each peaks represent 1 compound. The biggest, the tallest one is Syracuse. In the middle, here's the monos like fructose, like glucose, and this small one is the ribitol, which is our inner control. So another things I want to mention in this presentation is about the lab safety. Maybe you may mention that, you may notice that in the first step, the extraction, we use the chloroform as our non-pollen solvent, and the chloroform is very corrosive. Even just a few drops of chloroform can make this plastic foam place very ugly. And unfortunately, I got a few drops on my hands, and even I was wearing a pair of gloves, my hands were turned into like that. It was very itchy and painful. And luckily this strange feeling went away a few days later. So always be careful in a lab, no matter how familiar you are with the whole experiment. And I want to thank Dr. Chen, who helped me a lot in this project. Thank you for listening. Any questions or comments?
Great. Good job.
Nice job, David.
Thank you.
Questions for David? Are folks in Dr. Chang's lab continuing these experiments, David?
Oh yes, Dr. Chen and his group are still working on with the data. Yeah.
Nice. Hopefully they'll let you know what happens.
Yeah, I hope so.
Okay, excellent job.
Thank you very much.
Our next presentation is from Daniel Santander. Daniel is a computational biology concentrator, and let's congratulate him. We just heard recently he got a job as a R&D data specialist at Syngenta, which is in his home state of North Carolina. So congratulations. The title of the talk today is Computer Modeling for Greenhouses.
Cool. So thank you so much. Like Dr. Scanlon said, I'm going to be presenting today about computer modeling for greenhouses. This is my honors thesis, and I got the opportunity to work on it with Dr. Madsen and one of his former postdocs in his lab, who is Dr. Kale Harvick. And so it was a really cool project to get to work on with the two of them. Sort of the big idea of it is, in a traditional greenhouse, you have obviously supplemental light being hung, but sometimes it's hung at arbitrary heights and distances and things. So in this image, you can sort of see these are supposed to be rows of crops in a greenhouse, and you generally have like this hot spot in the middle of the greenhouse and some of the cooler spots on the outside of the greenhouse. And so these are areas of like low luminance, and whereas the center of it will be very high luminance. And so this is generally a problem just because, as you can see, the plants on the outside are getting less light, whereas the ones on the middle of the greenhouse might be getting too much light. And so the idea of the project was sort of just to optimize it and make it as uniform as possible. So go from this traditional lighting plan over to this optimized lighting plan, where the idea is just to have all of the plants in the greenhouse getting the same amount of light. And so if you were going to listen to my presentation, you could stop there, and that's sort of the big idea. But what is the motivations for this? So like I said, supplemental lighting can create hotspots in the greenhouse. And this is really a problem in greenhouse research. It biases the research, and sometimes you have to like move around your plants to make sure you don't have an extra variable in there. And it can also result in variable yields, where, for instance, the plants in the middle of the greenhouse might produce more fruit, they might be larger, things like that. And the ones on the outside of the greenhouse might have lower yield. And so the motivation for this was that in order to get more consistent yield, more consistent data throughout the greenhouse. But also we wanted to work on it with a computer program because it is really tedious to do something like this by hand. And so the computer program that we wanted to develop was something that could take in the type of light that you're using in the greenhouse, the size of the greenhouse, and the crop that you're growing, and it would return the optimal location to hang your supplemental lights. And so what does this look like in the real world? I apologize, I know these images are a little hard to look at, but the big thing to take away from these is that in a traditional lighting plan, you normally just hang your lights in a row. They're arbitrarily hung at the same height and at the same distance, and you might come in with a light meter, but generally it's just sort of you hang it right above the plants, and that's the intuitive answer. But in the other image, you can see in an optimized lighting plan, you can have lights at different heights, lights at variable distances, some lights can be turned in different directions, and so this would be something that would optimize the uniformity of it. And yeah, so how did we go about developing a model to do this? So it started with gonio photometric data. Goniophotometric data measures the light at different angles, measures the light intensity at different angles from the light source. So gonio means angle, photo means light, and then metric means to measure. So it's measuring the light at all the different angles from it. And so this was useful because we could develop goniophotometric data from any light source. So if we wanted to try different types of supplemental lighting, we could produce the same files and load it into our computer model. And also it sort of created a 360-degree 3D image of light intensity. And so this is where we started the computer model. And so we built up from this goniophotometric data. We started here and we ended up with something like this, where we could represent multiple lights in a greenhouse. We could put them in rows, we could put them in different positions and things like that. And so these were sort of, I'll go into a little bit more depth about how we got there from this data to sort of a full-blown computer model. So it started with loading up the goniophotometric data and then interpolating between the data points. So you can see there's really only about 360 data points in the goniophotometric file. And obviously we want to know any point. So the first step was to interpolate between the data points so that we could sort of estimate the luminous intensity of any point along this sphere of an equidistance from the light source. And these units were reported in candela, which, if you're not familiar, it's a photopic light intensity. It's not really useful to growing plants, but it's what they report the data in. So from there, we moved on from using luminous intensity to illuminance. And this changes the data from spherical values to Cartesian values, and it allows us to be able to calculate the illuminance of any point in the greenhouse. And so obviously we started here with the sphere, and now I arbitrarily sort of chose this bed in the greenhouse of points that we could calculate, but you could really calculate any point. It could be an inch away from the light source, it could be 10 miles away from the light source, it could be in the greenhouse, or it could be in a different country. You could calculate the light intensity or the luminous intensity. And so this changed our units as well. It brought us from candela to lux. And this is significant as we move on to the next step, which was converting to photosynthetically active radiation. Like I said, we were interested in growing plants, obviously, and we needed units that represented the spectrum of light that plants use. So the last step was to convert from lux to photosynthetic photon flux. And then eventually we were able to start producing these heat maps of sort, where we could see any point in a greenhouse, how much light is it getting simply from this one light source. And then from there, we wanted to combine different light sources. Obviously, a big greenhouse has a lot of lights and a lot of space. So we took this one light and we wanted to see how they interact well with different lights. And so you can see this model has about 6 lights in it. And even though there's a hot spot right underneath the light, they begin to develop an even bigger one through the middle of the greenhouse as they all interact. And so this was significant as well because lights are sort of a compilation of effects. And then for there, we went into the computational optimization. So now that we had a model to display any type of lights, I could choose the position I wanted to hang my lights at or anything. I wanted to go from having this and then computing my traditional lighting plan to an optimized lighting plan. So how did I go about optimizing the light? So really what you had to do was test different combos of lights to find the one that produces the optimal uniformity in the greenhouse. And this uses a lot of different computational techniques, mainly just linear programming. And this is challenging because it's something called an NP-hard problem. NP means that it's a non-deterministic algorithm that's solved in polynomial time. And so this means that you could have different results on different runs of the algorithm. But most importantly to note from that is that it's solved in polynomial time. On this graph to the right, you'll see that polynomial time is over here in the red. And as you increase your data size, the time for computation increases really poorly. So the more data you use in this computational optimization, the longer the computation is. Additionally, a solution can't be fast and correct at the same time. So there's sort of this trade-off. Do I want a really refined, correct answer, or do I want to wait a week for it? So this was the challenge of it. And of course, this became a real world problem as you begin to think about how to apply the software. As you begin to optimize larger greenhouses with more lights, it's obviously going to take a longer time. And so my conclusions, my research look to identify the optimum position to place the lights based on the crop, the type of light, and the size of the greenhouse. And future research would ideally look to optimize for growing in beds, not just the entire greenhouse, so rows of it, and as well as integrate software for crop interception models. So obviously plants are dynamic, they're constantly growing, they're not just on the base of the greenhouse. You would want to know how is the light changing as they grow. And then additionally integrate climatic data to know how natural sunlight affects the uniformity of light in the greenhouse as well. And I'd like to thank or acknowledge at least Dr. Cale Harbeck right here with some cool plants, and of course Dr. Matson as well with some cool plants. They really helped me out a lot with the project, and I'm forever in their debt, so thank you very much. Any questions?
So Daniel, that's a really cool study, but I'm going to put you on the spot with a question here. Why did you choose to make your light measurements using units of lumens or lux?
Yeah, so I actually didn't choose to. We got the data from Intertech, which is this company, I think somewhere in upstate New York, and they give us the gonio-photometric data. Unfortunately, I had no idea what it was before I started working on it, and that was just the units they gave me was in Candela. And so it was a really challenging process. It's actually the hardest step for me was to transition from Candela to PAR. It took me months to figure out the math behind it, really.
Yeah, because essentially there's a weighting curve that's used for
the luminous.
Basically has zero intensity at 400 and 700 nanometers. So
yeah,
it makes it a little bit difficult to work with. For example, if you use different types of lights.
Yeah, of course, it's different for every single light, which was why it was challenging. I had to develop now a second data set for each one.
Got you. Okay.
That's a really intuitive question. I think only you, Dr. Owens, would know that.
And Tom, I'll chime in. That's kind of the state of the horticultural lighting industry, is that they're reliant on this data taken for human lighting, and then they have to take spectroradiometer measurements to do lighting source specific equations. So
yeah.
Yeah. Daniel, I was curious if you had more thoughts about, you alluded to this, is that a crop canopy grows over time?
Yes,
so as researchers, how should we report light levels at crop canopy? Should we remeasure those every week or something and take averages over crop cycles?
Yeah, of course. So let me go back to an image and I'll give you an idea of it. So in this image, you can see I sort of have these calculation points in the bed of the greenhouse, and these are what I use to optimize. But ideally, you could sort of develop this like computer model of what the plants look like, the heights of them and different things, and then optimize based off of that. So it really just come down to building like an image of the plant in this three-dimensional environment, and then using these calculation points off of that. It would be, I guess, involved like a dynamic growth model of the plant, and then just putting it into this computer model.
That's great, thank you. And I have to chime in, I don't know of anyone commercially using this kind of method to optimize lighting layout in a greenhouse. So this could be a huge thing both for researchers as well as for commercial growers, especially if you're in kind of rectangular greenhouses with lots of perimeter space. In kind of really massive greenhouses with lots of central space, it may not be as much of an issue. But.
Awesome, thank you. Cool. Any other questions?
Beautiful job.
Awesome. Thank you all so much.
Thank you.
Great job, Daniel.
Okay, our next talk is from Jiaoqi Wang. Jiaoqi is a systematics and evolution concentrator. His plans after graduation are to attend graduate school. And today's talk is environmental effects of Zamia integrifolia, leaflet morphology.
Hi everyone, my name is Jiaoqi Wang. Today I'm going to introduce you to my research environmental effects on Zamia integrifolia leaflet morphology. So let's first of all have an introduction. Let me refresh your memory about the Zamia family. So Zamia is a genus within the family Zamiaceae. The sister group to Zamiaceae is the Cycadaceae, and they both are resides in the Cycadales, the order. So in case you haven't seen Zamia before, I took a picture in the LHV conservatory. It is exactly the species that I'm studying, the Zamia integrifolia, so you can have a picture in mind. Zamia are native to the Western Hemisphere from Mexico to Bolivia and Brazil, as well as in the West Indies. Here is a map of the distribution of Zamia. There are 79 accepted species, but different people have different opinions on that. The colors here are showing the major clades in the genus Zamia, except the blue star, which is the sister group Microcycus. I put a red circle in the map to show the populations of Zamia integrifolia that I'm studying is mainly distributed in the Florida state. So before I dive into the method and material, let me have a brief explanation of why I'm studying Zamia and why Zamia integrifolia. Of course, it's a project of a postdoc in the lab, but officially the whole family Zamiaceae, or even the whole order, I think maybe are mostly endangered species that are mostly defined by their leaflet characters. So as gymnosperms, they don't reproduce often as angiosperms, and Zamia integrifolia is one of the most wildly spread species in the family, and they have good herbarium records. So we're going to look for the correlations between the leaflet morphology and the local environmental factors. So first, for the material and method, the first data set I have is a collection of 127 herbarium sheets information. So on the right, you can see an example of the herbarium sheets over the overall looking, and at the red corner, you can find a paper label, and here is a close-up of the label. You can see the state, species name, description of the location, the county, collector, and date. So what I was doing is inputting the data into a computer file. That's the first data set I have. So the second one, the second data set I will do is to measure measurements of the leaflet parameters. As you can see here, the length of the leaflet, the width, angles, and the length of the internodes. Here I posted several pictures of the leaflet to show you the variations among within the species. So the third data set is the local climate of each collection. So I will pull that data sets from this website. It's all about the climate. You can see rainfall and temperature, a lot of interesting things. So for the results, right now I have a population distribution map. They're mainly distributed in the Florida county, as I said before. For the future work, I will take measurements of the leaflet morphology and use some R language to combine those data sets and analyze them. For the final step, we will look for the correlations between leaflet morphology characters and environmental characters. And if the answer is yes, environmental can have an impact on the leaflet morphology, we'll continue to ask if affecting the size or the shape of the leaflet. But if the answer is no, it is still a positive answer. We then can say that, okay, leaflet morphology is a good way to define different leaflets in the family. And with that, I would like to acknowledge the people in the Spec Lab. They are all helping me a lot, especially Dr. Sheila Shelsman, who is a guide, the mentor of my project, and Dr. Chelsea Speck, the PI of the lab, and also my mentor in plant science. Here are the references, and with that, Are there any questions? And thanks for your listening.
Nice job.
Nice job.
I could, this is Chelsea. Hi, Joshi,
Hi.
very nice job. I hope all is going well for you. What might you, thinking, with all the data that you were able to collect, what might you anticipate you would see as a trend across the ecological gradients that exist in Florida? Are there any patterns that you're sort of anticipating will emerge?
Right now, I haven't looked into the climate, but I would guess that if you have more rainfall, you will have a broader, I believe, a broader leaflet. Would it be true? That's my guess.
Mm-hmm. So you expect rainfall. So again, it's sort of the drought patterns corresponding with maybe different
Right.
aspects of the broad leaflet morphology.
Yes.
Great job, Joshi. Thanks for staying up so late.
Yeah.
Thank you.
Yeah, thanks. It's good to hear you in person.
Joshi, unmute your video for a minute so we can all see you and wave good night. Joshi is in China.
Okay. I haven't used this function
Hi. Hi, it's good to see you.
Great to see you.
Looks like him.
Yeah, it's really him.
Okay.
Sorry, I won't see you at graduation.
Yeah.
Thank you, guys.
Great job. Thanks. Our next talk is from Ziqin Wei, and Ziqin is a plant breeding and genetics concentrator. And after graduation, he will come back to Cornell and do graduate work in plant biology. The title of his presentation today is Genetic Basis for Carotenoid Stability in Arabidopsis thaliana. Yes?
Yes, we can.
Okay, thanks, Mike. And hi, everyone. I'm Ziqin. Today, I'm going to talk about my research at Dr. Lee's lab, which is about carotenoid stability in Arabidopsis thaliana seeds. First of all, I would like to introduce some background knowledge about this project. Carotenoids are a group of important natural pigments, including more than 1,100 members. They can be found in animal, plant, and fungi. They are very beneficial compounds for human health. For example, anthocyanin is great for reproductive health, and provitamin A is related to eye health. Most of carotenoids can serve as antioxidants, and they could also reduce age-related macular degeneration of eye. Although breeding efforts to enhance carotenoids content in crops has made many good examples, such as golden rice and golden maize, turnover and post-harvest storage cause great loss of carotenoids. To solve this problem, we firstly need to know two questions about carotenoids. The first one is where did carotenoids synthesize? And they are synthesized in plastids in plant cells. As you can see, there are mainly 5 kinds of plastids in plant cells, and they vary in carotenoid content and the way they sequestrate carotenoid. Another important question is the biosynthesis and the degeneration of a very crucial compound in carotenoids, which is beta-carotene. We try to enhance the stability of beta-carotene by manipulating 4 genes. The first one is PSY, which is a rate limit step in this pathway. We overexpress this gene in Arabidopsis seeds. The second one is BCH, which will cause the degeneration of beta-carotene and help with turnover. Thus, we use CRISPR-Cas9 system to knock out this gene. Another 2 genes are OR and HJJT. OR could stabilize PSY protein, and HJJT is related in vitamin E biosynthesis pathway, and HJJT overexpression could enhance vitamin E content, and which is assumed to be effective in helping with stabilize carotenoids. So for the strategy and method part, later I will introduce the plant materials used in this experiment, and we use HPLC method to detect the carotenoid content. And for the plant materials we used, we have 40 lysyl BP group. As you can see, each letter represents a manipulation to our plants. So we have 40 lysyl BP group, and then we add the operation to OR gene, and we also have 40 lysol BPO group. Also, we have 40 lysol BPOH group. So after screen for positive transformants, and we finally have 10 lys for each group. And after 8 weeks post-harvest storage, we have formed the seed germination results and carotenoid content results. So for the seed germination results, as you can see, the blue lines represents the wild type blank control. So as you can see here, so most of our BP lines and some BPO and BPOH lines have the same higher seed germination rates than the wild type. And as there are some a little bit lower, but we could assume that there are no evident difference between those lines and wild lines by comparing their data. And for the carotenoid content, we could see that carotenoid content basically matches our expectations. As you can see, the carotenoids are most in the BPOH line, and the BPO lines are second, and BP lines are third, and they are all higher than the wild type. And as we add operation to our plant materials, and as you can see, there is an evident difference between those two lines, and we can conclude that it is helpful to manipulate these 4 genes in this research to enhance the stability of carotenoids. So from the results, we could conclude that fitness of transformant plants is nearly the same as a normal wild type plants. And by properly manipulating these 4 genes, we can effectively enhance the total carotenoid content in plant seeds. And carotenoids in transformant seeds has a higher stability. So I want to thank Dr. Li and Dr. Tianhu Sun for helping me and guiding me through this research. And this research is also supported by Cornell and Robert Hawley Center of USDA. Thanks for your attention. Any questions and comments?
I have a question with regard to the CRISPR treatment that you did to knock out the enzyme that's involved in breaking down beta carotene in the formation of xanthophylls. So okay, was that gene was knocked out throughout the whole plant?
Also, we knocked out this gene in seeds, and after selecting for transformants, and we believe that it is knocked out in our selected lines for the content test.
The reason I'm asking that is because the xanthophylls that are downstream from that enzyme are absolutely critical in the function of photosynthesis. So those plants wouldn't do very well if you grew them in excess light.
Yeah, so that's the reason why we want to test their seed fitness to see whether we knock out this gene will affect their fitness.
But you said that it didn't affect their fitness?
Yeah.
That's very surprising.
Yeah, from the seed germination test, and we could see that there are very slight difference between different lines, and all of the transgenic lines were knocked out with BCH.
So you're only evaluating seed germination, not plant growth, in the lines where you knocked out that enzyme.
Yeah, so I discussed with my mentors, and we haven't decided which is a better way to evaluate the growth fitness. So finally, we decided that maybe the seed germination will be a good index for indicating whether they are good or not. So, and because for the plants we use in this research, so they are growing greenhouse, and they nearly do not need to use their fungal protection system. So we decided that the germination rate is important for farmers for selecting their seeds. So we did the plant seed germination test. Yeah.
But related to Tom's question, you didn't notice any chlorotic effects on these, and these were null mutations, and are these gene families duplicated? Are their functions duplicated? You got a good result.
Oh yeah, so in our previous plan, we decided to have some pictures for the psyllic in the transgenic plants and the wild type plant to see whether there's a difference in color. But due to this virus, so we did not do this test. But according to our previous observation, and there is some difference in color between the transgenic lice and the wild type.
Other questions? Okay, great job. Hope to see you soon.
Yes.
Yeah, thank you.
Nice job.
You guys have done great. Our last talk before lunch is from Matthew Sarzanowicz. Matthew is also concentrating in plant breeding and genetics. And after graduation, he plans to go to UC Berkeley and study plant and microbial biology there. His presentation today is entitled Transgene-Free Editing of Crop Plants. Matthew?
All right. Can everyone hear me? Is this working?
Works great.
Sweet. All right. Hello, everyone. My name is Matthew Sarzanowicz, and today I'm going to be presenting on my research that I've been doing at the Plant Transformation Facility with transgene-free editing of crop plants. So this research is still ongoing, and it's been put on hold due to the pandemic. So I'm going to break it down into the 3 Ps, the premise, the process, and the prospects of this research. So the premise is to develop a method of transgene-free editing of crop plants to overcome prior regulatory constraints. So normally if you're using something like CRISPR-Cas9 to edit a genome, you will insert the DNA that encodes for the Cas9 protein, and then that plant will become transgenic because it will contain that DNA. And when you're talking about agriculture and trying to release a crop onto the market, this becomes rather problematic due to the immense regulatory costs. So we're attempting to develop a method to edit a plant genome with a directed edit without using any incorporated DNA. So why transgene-free editing? Why is this so important? So as I mentioned, there's a massive regulatory cost that comes to any inserted DNA. So traditionally, if you were to insert a gene, there would be about a $30 million price tag to go through the regulatory process in the United States. And as you can imagine, there are only a pretty small number of players that can actually afford that cost. So it excludes academic players, small breeders, NGOs, and the limit of potential stakeholders also limits our potential to develop new and better crops. So there are new horizons that are emerging with gene editing technologies that can open up access to this editing to more players. So the $1 million question becomes, can we create targeted edits without introducing foreign DNA? And the method that my lab is trying to use to address this question is induce direct controlled editing by introducing purified Cas9 protein and its gRNA template. So rather than introducing the DNA that encodes for Cas9 and then allowing the plant to synthesize Cas9 and edit, we want to insert Cas9 directly as a purified protein so that there is no DNA. So how do we go about doing this? There are 2 integral parts of this method. The first is to insert that purified protein into a plant cell, and that's done with a gene gun. And then the second is to regenerate that edited plant cell back into a full plant. So biolistics, or the gene gun, I will walk you through how this process works. So to begin, in box number 1 here, we take very, very small gold particles between 0.6 and 1.2 micrometers. So microscopic, you see it here as a dust, and we precipitate a Cas9 gRNA complex onto the gold particle. So we effectively coat the surface of this with protein. Traditionally with biolistics, you would use DNA and coat them on the golds, but since we don't want DNA, we're just taking the purified protein product and painting each of these gold particles with the protein. We then allow that to dry on these discs, and you can see here all of that gold residue. There's a nice powder. And then we load it into this machine. And this is the biolistics machine that actually will shoot those golden bullets into plant tissue. So this works by inserting these discs and then creating a vacuum inside of this chamber with your plant tissue. And then you can select the pressure that the gas will be introduced into the chamber that will fire these bullets into the plant tissue. And so we can choose between 350 and 2100 PSI, so quite a large range. And the higher the pressure, the more force those bullets will have, and the deeper they'll penetrate, but also the more damage that they will cause. And then finally, we'll have, you can see here, these are some wheat embryos, and you can see that there's some residual gold residue on the embryos. And so that's after it's been fired. And that's kind of the general biolistics process. But how do we go about optimizing this process? So there are 2 main ways. The first is, how do we optimize actually getting the gold into the plant tissue? And we use a GUS construct. So this is DNA, so it's just used to optimize. And essentially GUS is this marker that will turn blue when you expose cells that contain GUS to a substrate. So you can see here all of these tiny blue dots on the surface, and that's every cell that has received and successfully expressed GUS, and that you can quantify that. So effectively we can look at different pressures, different number of shots, different sizes of gold on a same type of plant tissue, and then quantify the number of blue dots. And the more blue dots we have, the better we're doing. So we can use this to kind of figure out what is the best combination of variables to just introduce the gold into the plant tissue. And then the second is we actually want to monitor how well are we editing. So to do this, we use, we insert Cas9 with a guide RNA that edits the PDS gene. So PDS is phytoene desaturase. It is the gene kind of at the top of the carotenoid biosynthetic pathway. And when we knock it out, a plant is no longer able to produce carotenoids. So when you expose a plant without carotenoids to light, it will become photobleached and they appear as albino. So we can try different methods of incubation or light versus dark treatment with the Cas9 inside the plant to monitor how well the editing is occurring. And we compare the ratio of albino plants to green plants that are regenerated. And the higher that ratio is, the better we are doing in terms of editing. So you can see here, this is kind of a mass of plant cells. It's not differentiated, but this one appears to be starting to grow some leaf-like structures. And you can see that they are white. So it appears to be an edited leaf. And we can compare that to leaves that develop that are green and find the ratio between them. So we try to optimize for a higher ratio. But as you can see here, this is not a plant. This is just a blob. So how do we go from turning blobs into plants? And so that is what the main part of our work is tissue culture. So actually the process of turning single cells back into plants. It's a very finicky process, but throughout this presentation, you've seen all of these plants kind of growing in a gel-like substance. So that gel is agar, and inside of that gel matrix, there are a variety of major and minor salts, plant vitamins, amino acids, organic acids, and plant hormones. And all of these components work together to dictate how a plant grows, and we can manipulate them to change plant growth. So as an example here on the left, this is an aquatic plant that we were experimenting on in class. And it's grown, these are all identical media except for plant hormones. So we varied the ratio of auxins to cytokinins in the hormones. And as you can see, it has a tremendous impact on how a plant grows. But hormones aren't the only thing that, I'm gonna move myself 1 second. I can't see, here we go. Are not the only thing that control plant growth. So all of these components actually have a major impact on growth. As an example, this is potato growing in vitro. And I only varied the amount of sucrose. So you can see, and this is about 30 grams per liter of sucrose, root growth is robust. And if you double that, you get inhibition of root growth. And so it kind of follows a normalized distribution. So for tissue culture in general, every single one of these components, every nutrient plays its own role in plant physiology and development. And typically every species, actually not typically, always, every plant species has its own very particular needs. And then within a species, every variety. So when you're working even with 2 different varieties of rice, for instance, they will have their own media that they grow in, and they will not grow well in a different media. So a lot of this process comes not only with optimizing the editing procedure, but then trying to get plants back out from the editing. So what do these things look like when they're done? So I have some examples here that I've been doing. So this is a Chardonnay grape that I was working on right before I left. You can see it's rooting, it's doing quite well. These are soybeans that I worked on over the summer at a different research institute. They were doing soybean research, and then some apple that I was doing also at Cornell. One thing I want to mention is this research with transgene-free editing is particularly important for clonally propagated crops, so things like grapes and apples, because you can't do a back cross. So for soybeans, you could insert DNA and then do a back cross to a wild type to remove that transgene if you don't want a transgene in the final product. But if you want to preserve the characters of things like Chardonnay or an apple variety, you can't do any crossing. You must clonally propagate it. So any inserted transgene would be permanent. And so this method of transgene-free editing is quite important for clonally propagated crops, which is why we're working on apple and grape. So what are the future prospects and what are we going to try to do? So we want to continue optimizing gold particle size, the gun pressure, and explant tissue, which is the type of tissue that we use for transformation. And we also want to optimize our ability to select for transformed plants so that we can get as many edited plants out of the process as possible. And then we're also starting new research to test new technologies to facilitate plant regeneration. So the ultimate goal of this research is to produce an efficient, affordable, transgene-free editing platform, and then to democratize access to affordable plant editing technologies to the public sector. And with that, I want to acknowledge the team behind all the magic. So again, this research is done in the Cornell Plant Transformation Facility, and my PI is Dr. Matthew Willman. Thank you for your attention.
Great job, great job. Questions for Matthew?
Matthew, have you seen like anything on acceptance of the transgene-free edited crops? Have you like talked to anybody about it? It seems like it could be really beneficial.
Yeah, so there is one major product. So the company, I actually have it here, not for this presentation, I just happen to have it. So Calyx, which they produced actually the soybean, which is, it used to have a transgene. So the way they did kind of the alternative approach where you insert a transgene, edit, and then back cross to remove a transgene. And so they produced a high oleic soybean oil, and I actually have some here. So I worked there over the summer, and that's the first ever gene edited product on the market, and it's been selling quite well. It is not labeled as gene edited, so they labeled it actually, as we discussed in class, as non-GMO, which is kind of interesting. But due to the way that transgenics are regulated in the US, if a plant does not contain foreign DNA, it's actually considered to be non-transgenic, and you can label it non-GMO. So I think as of now, consumers, perhaps because they don't know, but they seem willing to consume it more so than perhaps other transgenic products.
Other questions for Matthew?
Matthew, can you hold that Calyx product up again?
Yeah.
Nice show and tell.
Yeah.
You can't really see. I have a different one too, but
wow, that's a big jug of oil.
this is a gallon. I know. This is like, I don't know what I'm going to do with this. I think it's more of like a, it's a memento.
Okay.
I don't know. Like, what do you do with a gallon of oil? It's like
the paperweight.
I think you fried donuts and cheese curds in it.
So when I was there, they made so many cheese curds with it, and they were incredible. I mean, the Minnesotan cheese curds, just incredible.
Except you really should fry them in butter.
Mm.
Not oil. Just saying.
Any other questions? Our first talk this afternoon is from Ho-Guan Yan, and he's concentrating in plant genetics and plant physiology. After graduation, his plans are, I quote, to survive the pandemic and then study at Rutgers University for graduate school. I'm sure that will happen. And the title of his talk is Locating the Genetic Locus Associated with Weeping Phenotype in Malus domestica, talking about apple blossoms.
All right. Hello, everyone. This is Ho-Guan Yan, and I'm presenting my research that I've been doing ever since last summer in Geneva. So some background information and like the overall goal of the research. So the reason why we did the research is we believe that understanding the apple tree architecture can help us to improve the uniformity of the apple trees, and therefore can potentially give us a higher efficiency in production by introducing some like mechanical harvest instead of just picking the apples by hand. So the goal of this research particularly is to locate and identify genes that are associated with the weeping phenotype in the domestic apple trees, which we will later refer to as the WD locus. So there have been previous efforts done for this on this direction on some other Rosaceae family species, such as peaches and crab apples, but none has been done in the domestic apple, and this is what we're interested in. So what is weeping phenotype? So as you can see here in this comparison here, the weeping apple tree is usually more dwarf, and the branches tend to grow laterally and downwards, compared to the center tree, which is like usually taller and the branches grow upwards. So it's pretty distinctive pattern, as you can see here. So the method that we used in this research particularly is that first we phenotype a half-seed population, which is the progeny of a heterozygous weeping population, open pollinated, and then we isolated DNA from each single individual in that population. And then we constructed two sets of DNA pools with those DNAs. One is the whipping pool, one is the standard pool. So each composed of 10 DNA from that respective phenotype. And we screen those DNA pools with various SSR markers, short simple repeat markers, throughout, like from various parts of the genome, in order to see whether there's a distinctive pattern between the two sets of pools to see to indicate whether there are potential genetic linkage or not. So if we see a potential linkage, we will further confirm it with other with more individual DNAs and more wider range of DNAs as well. And after that, we will see if the pattern is also true for the neighboring genetic markers as well. But if not, we're just going to keep repeating this process until we find a positive linkage. So the phenotyping result is pretty interesting. So here in this diagram, S stands for standard, SL stands for standard-like, which is the intermediate that's closer to the standard phenotype. W stands for whipping, WL is whipping-like. So what we can see here is that we have a number of intermediates, and that indicates the so the expression of this phenotype is not directly linked to one single gene. So it's probably there are other factors that can actually play a role in expression of this phenotype. So the result for the genetic analysis is, so this is the graph that I had for one of the marker. So those are from each individual line is from individual that's in the whipping pool or standard pool. So as you can see here, so this is the band that we're interested in. Which you can see here, 17 out of 19 in the whipping population that we tested has this band, apart from these two. And only 1 out of 19 in the standard pool that we tested has this band. So this is a very distinctive pattern that's been shown here, which indicates a very strong genetic linkage. And therefore, what we did next was so marker 2 was the marker that was in the previous slide. So what we did is we tested the marker that are neighboring to it. So marker 1, marker 3, marker 4. And what we see here, the recombination frequency is quite what we expected actually, and it's very beautiful. So marker 1, which is the upstream of marker 2, has a higher recombination frequency than marker 2, and which is 8.5%. And marker 2 has like 2.8%. Marker 3 gets even lower, which is like there's no recombination even present here. And marker 4, like as it goes further away from marker 3, it gets higher again. So this indicates that the locus of interest is probably very close to marker 3 here. Okay, so this genome region is in the chromosome 13, and in the 945 kb region, and also for the standard control, there's also a similar pattern, but the recombination event rate is actually higher, but it still follows the same pattern as observed in the whipping ones. Okay, so in conclusion, the WD locus, which is associated with the whipping trait in Malus domestica, has been mapped to a genomic region of 945 kb region on chromosome 13 of the genome. And based on the result from the phenotype, there could be other genes also that play a role as modifying effect. So for future research, what we have been doing is we're gonna have finer mapping of the region, and also we're gonna analyze the RNA sequence to determine like which exact gene is actually the causal one. Yeah. So big thanks to an SF grant, Cornell Architect Community, Summer Scholar Program, Canon Xu's lab, and my lab mates who helped me a lot during this research. Thanks for listening.
Question?
Job.
Thank you.
So are there dozens or even more genes in this 945 kb interval?
So I think from what I remember, we checked the genes in this region. There are like probably about 20-ish genes that are, yeah, within this region.
Nothing obvious like gibberellin hormone or anything?
Yeah, there are a few that are related to hormones, but we're not sure at this point yet whether they are actually the actual causal gene yet.
Oxen, I would think.
Yeah, we need to wait for the RNA-seq data to actually be sure.
Nice job.
Thank you.
Good. Okay, our next talk is from Lillian Trainer. Her concentration is in genetics, breeding, and pathology, so a mixed concentration. After graduation, Lily will begin an advanced track program through Ithaca College and Cornell to obtain an MAT in biology and agriculture education, so great luck with that. The title of her presentation today is Evaluation of Plenty Belize Garden-Based Learning Programs.
Thank you, Mike. Hello, everyone. This talk is based on a study that was designed and performed by Lily Cohen, Audrey Shutak, Sarah Scott Cruz, Martha Williams, and myself. To provide a little bit of background about this project, it took place in the Toledo district of Belize, which is in the southernmost region as pictured in the map at the bottom right corner of the screen. This region is reliant on subsistence slash-and-burn agriculture, pictured at the top of the screen. Due to this, in combination with an increasing population, the agricultural system is becoming unsustainable. This region is primarily Mayan, Garifuna, and Creole populations. It also has many impoverished and rural schools. The project we are evaluating is called the GATE project, which is run through Plenty Belize. GATE stands for Garden-Based Agriculture for Toledo's Environment. The mission of this project is to address environmental degradation and unsustainable agriculture and poor nutrition. The goals of this project are to create organic school gardens that are replicable by the communities and to promote organic gardening and sustainable agriculture. This project has developed programs at over 40 schools, though not all of these gardens have been successful. Cornell first became involved in this project through Marcia Eames Sheevely. In 2009, students traveled to Belize for the first time, and for many years after that, they traveled there to create gardens, give lessons to students, and train teachers on how to incorporate the curriculum into the garden. 2019, the year that I went, was the first year that students conducted an evaluation. This image was taken at Bob's Bible School, which is in the Toledo district.
Pronouncenames.com.
Goals of involvement include creating an unbiased evaluation and suggesting next steps for the project, as it was in the concluding phases and they were wondering what worked, what didn't work, and what they should do next. The top left image was at one of the first schools we visited, which had a very successful garden program. They allowed to harvest produce from their gardens, and then they served that to us for our lunch. Our methods included traveling to schools, conducting interviews, focus groups, and surveys. The top left image here is an interview that we conducted with Dawn, who was a member of the Plenty Belize board, as well as a member of the PTA for one of the schools that we were evaluating. Our interviews were primarily conducted with principals, teachers, and community members, while we did focus groups for students and women's groups. These interviews and focus groups focused on topics such as support and resources, nutrition, challenges, sustainability, relationships, and a few other topics that we identified as important for the evaluation. As you can see from the images on this slide, most of our interviews took place in either the garden or greenhouse setting. We provided surveys to younger elementary youth, and these surveys consisted of statements with a response scale of strongly agree to strongly disagree, check boxes, as well as some visual responses, such as what is pictured on this slide. The reason we chose these formats is because not all students were fluent in English, and most of these were easy to interpret. This would likely lead to more accurate responses. We did also include a few open response questions so that students could provide more detail if they were able to. Our observations refer to what we directly saw and heard when we traveled to the schools. Some highlights of our observations include that support for these programs has worked well, particularly the interactions and frequent site visits by Plenty Belize staff were valuable to the schools. We noticed how important it was for the curricular subjects we integrated into the garden space. This is because many teachers are already stressed by trying to meet the demands of standardized testing and don't see how they can fit one more thing into their day. Showing teachers how their current subjects can be incorporated into the garden space is essential and may contribute to helping them adopt the program. We also noticed that some schools were only addressing topics such as nutrition or science in the garden, while other schools were addressing those in addition to topics such as English and math. Using the gardens for more subjects increases the success of them and likelihood that teachers will adopt them. The bottom left photo here is a project that students did for recycling, and they managed to recycle bottles to build garden beds. Our interpretations refer to how we understood what we observed and how we connected it to a larger picture. Our highlights of our interpretations may be divided into overall support and the value of the program. Community support is essential, and ownership through participation of the community in a variety of ways was found to greatly contribute to the success of programs. Women's groups were an important resource for some gardens, and they benefit both the schools as well as the women because it provides them with a source of income and the skills to grow their own food. Parental support is encouraged by academic success. Some parents only see the value in traditional academic learning and don't understand how gardening fits into that. By demonstrating to parents how the gardens can be used to teach traditional subjects, they're more likely to support the garden program. Community partnerships were found to strengthen the sustainability of the garden by providing continual support despite possible turnover in staff at the schools. Additionally, we realized how important it was for the success of the programs to have the support of the staff in the schools. One Standard 4 teacher stated that teachers need to be invested. You can pump schools full of supplies, but the garden will fail if people are not invested. We discovered for the value that the garden involvement contributed to positive youth development such as belonging, generosity, independence, and collaboration. School gardens also contribute to food security for both the students in the school as well as community members. One teacher captured this well when he stated that what really amazed him was when a needy child was eating what they grew, and without this project, that child would have a hungry day. Other teachers commented on how much better behaved and better focused students were when they were not hungry because they were receiving nourishment through the garden program. From our observations and our interpretations, we developed a set of recommendations, which included professional development, community engagement and partnerships, a strategic plan, needs assessment, and further evaluation. For professional development, we decided to focus on teachers. Plenty Belize should hold workshops for teachers that explain why gardens matter and also convey garden knowledge such as pest management and composting. Hopefully, if teachers can see the importance of the gardens and the beneficial impact they can have, and also have the confidence to manage the gardens, these programs will become more independent and less reliant on support from Plenty Belize. The image on the right is a pest management system designed by students. They read that spreading grease on blue plastic would attract the bugs and then they would stick there, and this would keep pests away from their plants. The top image is a composting system at one of the schools that was not being used because many teachers do not know how to compost. We discovered that the community learning could be incorporated into the greenhouses and garden spaces. If the schools hosted community workshops on topics such as organic agriculture and the community was able to directly participate in the garden, this would likely increase their support for the program. Women's groups were a valuable resource because they could tend to the garden over school breaks when students could not come in. We also thought it would be beneficial for them to partner with colleges such as Bob's Bible School, which I mentioned earlier, which has a very successful horticulture program pictured here on the right. Students involved in this program could lead workshops for the school programs on topics such as building gardens and managing them. We discovered that many schools were not receiving the resources they needed, while other schools were receiving resources they did not need. An example of this is one school that needed seeds and another school that had plenty of seeds but needed better quality soil. We suggest that they develop an application to ensure that resources are going where they are most needed, and this will contribute to sustainability by using resources to their optimal potential. In the middle of the slide here is a picture of a school that was able to obtain its own resources and were using them to their full potential. And the top is a school that was given a lot of resources, but they weren't able to use all the resources that they were given. A strategic plan would enable Plenty Belize to solidify their core values, vision, and mission in order to determine how to best devote staff and board time and efforts. While the main focus of my talk is the study, it wouldn't be complete without discussing the story behind the study. This experience was filled with challenges, such as that some of the people we interviewed spoke little or no English, and a few participants did not show up to our interviews. Additionally, for me personally, it was a challenge to adapt to the environment, food, and housing conditions due to a lack of things that I would consider necessities, such as electricity, running water, and closed sleeping spaces, and dairy products. I was also bitten by so many ants that I thought I was going to lose my mind. Despite these challenges, this experience was filled with many adventures such as breathtaking nature, cultural immersion, the opportunity to fly a plane as pictured in the top right corner, and so much more. I would like to offer a special thanks to our faculty mentors, Marcia Eames Shively and Fiona Doherty, who were so supportive through this process and absolutely instrumental to the success of this evaluation. And that's all I have, so what questions do you have for me?
Great job, Lily.
So Lily, can you speak to the school garden movement in the United States? I know for a while there was a lot of interest. California was going to mandate that for all the school districts. Is that still sort of an initiative around the country?
So I'm not familiar with what's going on in California. It's definitely increasing. I know even in my home school in New York, they recently developed a community gardening project in collaboration with the schools. And I'm the New York State coordinator for the National Junior Master Gardener Program. And so through that, I'm kind of connected on the national scale to what's going on. And I'm definitely seeing an increase and more interest in this. And I think one thing like that the NJMG program is doing is that they're developing school curricula in coordination with state mandates so that it can be implemented into the garden. And I think that is really contributing to these programs being adapted in the US.
Great, thanks.
Okay, great job.
Thank you.
Thanks, Lily.
Nice job, Lily.
Our next talk is from Isabella Magna Iannuzzi. Her concentration is plant breeding and genetics and applied plant science. After graduation, Isabella will be working for Professor Kara Cox and then applying for admission to graduate school in the fall of 2021. Her title of her presentation today is How Do We Test Thrips Resistance? Isabella. See your slides.
Okay, perfect. So hi, like Mike said, my name is Isabella Magna Iannuzzi, and today I'm going to be talking about the research I conducted in summer 2019, looking at how we test thrips resistance. So I briefly want to talk about what thrips are. So they're small insects. You can see over here the larvae and the adults. They're in the order Thysanoptera. And in this particular study, we looked at onion thrips or Thrips tobacci. And the reason we looked at them is because in New York State, which ranks 7th in national onion production, they can cause between 30 and 50% crop losses. And onion production in New York State is also valued between $40 and $50 million. So they can cause extreme economic distress for farmers across the state. We had 3 main objectives for this study. We wanted to understand how insecticide exposure affects thrips mortality. We had a contact assay, a feeding assay, and a contact and feeding assay in addition to a field simulation we designed. We wanted to determine which assay most accurately represents field rates because we wanted to create a link between laboratory experiments and actual field conditions. And we wanted to discover which assay was the most simple to execute. So if we want to make these sorts of recommendations to crop consultants and other scientists, we need to be able to explain how simple and easy the methods were to actually execute. So we looked at 4 bioassays in the study, the first being TIBS, which stands for Thrips Insecticide Bioassay System. And this was a contact-only assay where we coated the inside of different test tubes with different pesticide concentrations, left them to dry, and then incorporated the thrips. We then looked at a feeding assay where instead of through contact, the pesticide was introduced through the feeding solution, so the thrips were only coming into contact with it through ingestion. Our third bioassay was a leaf dip, which was a combination of a contact and a feeding assay where we took cabbage discs and soaked them in different pesticide solutions. We then dried them off and introduced them to these petri dishes with thrips. And the reason that we used cabbage discs instead of onion leaves, because we were looking at onion thrips, is because onions have pretty thin and small leaves, and we wanted to ensure that there was consistency throughout the different replications of this bioassay. Lastly, we designed and conducted a field simulation where we grew onion plants in the greenhouse and then separated them into these different groups and applied different pesticide concentration sprays. So the results of this experiment were super interesting. The feeding assay over here on the bottom, the leaf dip in the middle in magenta, and TIBS up here in blue. This is the log dose mortality curve with mortality on the y-axis and log dose in parts per million on the x-axis. And the LC50s, which is the concentration at which 50% of the insect population was dying off, was statistically different for all three laboratory bioassays, which is very interesting because all three of these are readily used in the literature. And you may notice that the field simulation is not on this graphic, and that's because when we used our probit analysis with the data from the field simulation, it did not generate a curve, unfortunately. So our feeding assay had the highest LC50, which means it took the most amount of pesticide to kill off 50% of the population, and is interesting because it shows that contact is a really important part of the pesticide's mode of action. And this idea is further supported by the fact that the leaf dip had the smallest LC50 and TIBS had the highest mortality at low concentration. So where contact was incorporated into the bioassay, the thrips were much more sensitive to the amount of pesticide being applied. Takeaways from this study: the feeding assay was by far the easiest to set up. It required the least amount of resources and time, and it was very easy to check on the thrips and assess mortality. And again, suggests that contact is an important part of how pesticides work at killing off thrips populations. TIBS was definitely the most complicated. It required a lot of time, a lot of resources, and a lot of waiting also, because you have to make sure that the pesticide is coating the inside of the test tube effectively. And again, we would just need more replications of the field simulation in order to definitively conclude which laboratory bioassay most accurately represents field conditions. I would like to thank Cornell Agritech and Cornell CALS and the Geneva Summer Scholars Program for funding this project, and Juan Silva for the code for the probit analysis we used to analyze this data. It was extremely helpful. And I really want to thank Erica Moretti and Brian Nault, who were instrumental in helping me come up with this project and supporting me throughout. These are all of my resources, and I'd be happy to take any questions you may have. Thank you.
Good job.
Beautiful job.
Questions for Isabella? Do you think it would be difficult, Isabella, to eventually turn one of these into something that could be done quickly so that it could inform growers' spray practices?
Yes, I do. So I actually gave this talk yesterday to the entomology department, and I had 10 minutes, so I was able to explain a little bit more. But even though the field simulation didn't fit our probit analysis, the LC50 it generated was most simple to that of the feeding assay. So that's what we would recommend right now, just because of the information and the results we have from the study. But that's what we're hoping, is that there's a quick and effective way for everybody, the growers, extension agents, farmers, to check their thrips population and test the resistance levels.
Excellent.
Other questions, comments? Okay, beautiful job. Thank you very much. Okay, our next presenter is Lingwei Wayne. Her concentration is plant pathology, and after graduation, she'll continue that, studying right here at Cornell in PMB. And her talk today is entitled Molecular Differences Between Tomato Immune Receptors FLS2 and FLS3. Lingwei?
Hello guys, I'm Lingwei Wang. Today I want to talk about the project that I did last summer in Martin Lab about the molecular differences between tomato immune receptor FLS2 and FLS3. So the project is about the tomato PST pathogen. PST stands for the Pseudomonas syringa passiva tomato. It is a bacterial pathogen which leads to the bacterial speck disease in many Solanaceae species, including tomato. It causes a huge loss in the tomato production in New York State. The pathophage then serve as a model for studying bacterial pathogenesis and the plant immunity. The effort of the project is to understand the interaction in the molecular level, which can help us to better understand the plant immunity and possibly help us to find new resistance against the pathogen, which can help us to control the disease. And FOS2 and FO3 stand for flagellin sensing 2 and flagellin sensing 3. The reason why we're interested in this topic is that both FOS2 and FO3 play an important role in plant immunity, and they are similar in many ways. However, the individual contribution to the resistance is still unknown to us. So they are both pattern recognition receptors and response to different peptide on PST flagellin, so which is flagellin 22 and flagellin 228. And they are both receptor-like kinases, which means they regulate the pathway through phosphorylation and give you a mindset that they have the property as a kinase. And upon the recognition of flagellin, the immune response in plants will be triggered, including the ROS response. ROS stands for the reactive oxygen species. A rapid and transient burst of ROS is a key response of plants, and in the lab condition, we can detect its production quickly with the treatment of, in our cases, flagellin 22 and flagellin 228. So the more ROS production could be an indicator of higher immune response. And the start of our project is a very interesting observation in our ROS data. So the x-axis stands for the time after we add in the peptide, and the y-axis is our ROS production. As shown here, the ROS response for FOS3 is more sustained than the ROS response of FOS2. If we expand the time a little bit, FOS2 will respond for about 100 minutes, while FOS2 will only respond for 60 minutes. And the structure of the receptor as shown here, including 3 domains, so which is the kinase domain is inside of the cell, and the transmembrane domain and the lucent-rich repeat domain. We hypothesized that the kinase property of these 2 receptors contribute to the ROS difference. So we align the sequence of the kinase domain and find that FOS2 and FOS3 share a common lysine, which involving ATP binding process is an important step in the phosphorylation. And FO3 share a common glycine-rich motif, which is GXGXSG motif. This motif has been shown to have an enhanced kinase activity in other kinase family. So we hypothesize that this motif will give enhanced kinase activity to FO3 as well, which could possibly be the reason for the difference in ROS response. So we built 4 different constructs to test our hypothesis. First, we replace the lysine and lead to a loss of kinase activity in both FOS2 and FO3. We use it as a negative control in our following ROS assay. And then we replace the glycine-rich motif in FO3 with its counterpart in FOS2, which is GXSXSS motif, and make FOS2 mutate to have a glycine-rich motif. The kinase assay support our hypothesis that the glycine-rich motif did result in an increase in kinase activity. So the next step is to see whether the increased kinase activity is the reason for the different ROS response. These 2 figures here show the total ROS production of our 4 construct and the wild type. So the loss of kinase activity in these 2 constructs did show a loss of ROS response. Surprisingly, the FOS2 mutation result in a decrease of total ROS production rather than an increase. For the FOS3 mutation, we observed no significant effect of the mutation on the total ROS production. So the results suggest that although the glycine-rich motif influenced the in vitro kinase activity, the differences in kinase activity alone does not explain the differences in the ROS output between these 2 receptors. It is currently unknown to us why the in vitro results do not translate to the implanted ROS assay, but future experiments studying the kinase activity in vivo will help us uncover the mechanism of kinase activation in FOS2 and FOS3. And the project as a whole suggests no single domain of FOS2 and FOS3, including the LRR domain, transmembrane domain, and kinase domain, can explain the sustained ROS response in FOS3, and multiple structure features unique to FOS3 might contribute to the sustained response. And it's currently unknown whether FO3 can function in species outside of Solanaceae, but future efforts to express FO3 in more diverse species could determine whether Solanaceae-specific protein partners are involved in the FO3 ROS response. And in the end, a lot of thanks to the PI of the lab, Rick Martin, and the member of our project team, Robin Roberts and Alex Phil, and other lab members who helped me during the summer. And here's my reference. I'm glad to take any comments and questions.
Job.
Great job. Good job. Lots of time for comments. Okay, beautiful job. Very sophisticated.
Thank you.
Okay, our next talk is from Jessie Zhou. Her concentration is computational biology. After graduation, Jessie plans to go get a job from IQVIA, which is a pharmaceutical manufacturing firm in China. And the title of her talk today is Maize Genome Prediction with Human-Based RNA Splicing Models.
Hello everyone, today I'm going to talk about the maize genome prediction with the human-based RNA splicing model. First, I'd like to send my thanks to all members of the Berkeley lab. I've worked at Berkeley's labs in the summer of 2019. The lab group has developed a wide range of data, bioinformatics, and molecular tools for plant genetics and diversity studies. The boss of our lab is Edwin S. Berkeley. He's the research genetics with the USDA ARS, the adjunct professor of plant breeding and genetics at Cornell University, and he's also a member of National Academy of Sciences. To begin with, I'd like to briefly introduce our lab tradition, the hackathon. Basically, it's an annual activity that all lab members and traveling scholars work together for a certain project. And the project for 2019 was studying KP. I was in team 2, and our team focused on studying KP models. Our team leader is Catherine, and she's an expert at building plant models, and she really taught me a lot of things, and I want to send my special thanks to her. So first, what's KP? KP is an IPA and a repository of ready-to-use training models for genomics. It offers a quick and easy way to share these models, no matter you prefer using R, Python, or command line. When we checked the model tool, we found that the most popular tags in KP is the RNA splicing tag. So we decided to take a closer look at this one and to find out like why RNA splicing is so important. So first, what's RNA splicing? Basically, it's a newly made mRNA transformed into a mature mRNA. During this process, introns are removed so that the exons can connect it with each other. Due to the time limitation, the RNAs, I would just introduce one RNA splicing model here, and it's Optimus 5', and it's human-based, which targets the 5' untranslated region. 5' UTR is the region of an mRNA that's directly upstream from the initiation codon and is primary determinant of translation efficiency. In other words, it's quite important for RNA splicing and also the translation. So here comes the question, what if we predict the maize with a human model? The reason we do so is that there's no plant-based model, plant-based RNA splicing model on KP, and also the Optimus 5' seems to be robust, like according to the high accuracy that mentioned in its paper. And also, if the model can help, we can then build an RNA splicing model for plants using this model. And if it cannot help, then here we found a gap. So what we do first is that we go to the maize GDB, gathering the maize genome files, and try to compare the prediction result with the human chromosome file provide from KP, and we submit our Python script on the Bitbucket, and here's the result. This plot shows the cumulative distribution of MRLS of Optimus 5', and we can see that maize here has a sharper distribution curve, and the maize also has a higher mean, median, and max comparing with the humans. However, the Optimus 5' paper suggests that the mean and median MRL should be like around 7, but all these they're just around 5 and 6. So this is not so ideal. Then we decide to take a closer look at the prediction. We use IGV to visualize the results, and the upper tab is the reference genome, maize reference genome, and the lower one is the prediction result of Optimus 5'. And the first figure shows the correct condition. It's that the Optimus 5' did find the 5' UTR is at the right position, and the length is also very good. And figure 2 shows that it didn't find the correct 5' UTR because the length is like too long for the 5' UTR, but it got the right position. Here I just choose 2 representative results, and the whole visualized prediction of maize 5' UTR is very messy. So it suggests that the 5' UTR have a kind of low accuracy in finding the maize 5' UTR. So the gap did exist, and the human model cannot predict, cannot be used for prediction for maize genome. And this is the references. And thanks for attention. And so questions.
Good job.
Okay, well, thanks very much. That was great. Our next talk is from Martha Williams. Her concentration is in ecology of managed landscapes. And after graduation from Cornell, she will attend NYU Steinhardt's MA Food Studies program, part-time in the fall. The title of Martha's talk is Garden-Based Learning at Cornell Cooperative Extension, Nassau County.
Hello. I hope that everyone is doing well, and thank you for the introduction. So this is actually about my 3 consecutive summers at Cornell Cooperative Extension in Nassau County. So first I'm going to give some definitions, then I'm going to tell you a little bit about the place where I actually worked, and then I'm going to talk about the garden-based learning programs, some challenges, and outcomes. So what is Cornell Cooperative Extension? So Extension actually exists throughout the country, land-grant universities and colleges to provide non-formal environmental and agricultural education. So in New York State, that's different extension offices in each 62 counties that is really focused on helping New York State families and communities thrive. So Cornell Garden-Based Learning is another way of really achieving that mission and those goals within it. So the focus of my work was really to engage, to empower, and provide relevant experiences to the youth that I was teaching. So East Meadow Farm. East Meadow Farm is where I did the most of my education. I'm teaching and are there again to promote the CCE mission. They have demonstration gardens like you can see up here. And they also had a farm stand on Fridays throughout the summer and also throughout the falls. And right here is actually some of the produce that we actually collected during that time that were grown by the Master Gardener volunteers, which is just a group of people who come and volunteer and get a lot of expert knowledge on gardening and growing things. They also have classes, they have plant diagnostics. Right here is an example, is one of the farm animals there. Her name is Tilly, and she's also really evil, so you can't trust her. Okay, so the garden-based learning programs. So I did Seed to Salad in 2017 summer, and then I did Junior Master Gardeners for 2 consecutive summers. And then I also did a lot of one-off lessons and just gardening basics. And right here is actually an example of some microgreens that a group was growing for one of those lessons. So program objectives for Seed to Salad. So really the focus of this is to utilize small space, which actually right here down on this picture down below is actually a picture of something growing in a small space garden, which is one of the demonstration gardens at East Meadow Farm. And we actually use that as one of the models for creating the smaller space garden. They gained the basic horticultural skills. They were able to engage in multidisciplinary activities, meaning that they were able to engage in things that had to do with English and writing and things of that nature. So it's not just purely science like when people think about when they hear garden-based learning or really any other type of really like gardening focused curriculum of any sort. So up here in this left photo up up top is actually one of the things that I created for this group that was doing the Seed to Salad program. So it was actually a way for them to interact more with something physically in front of them that they can write. So there's something like learning about gardening problems, they would draw something like, what does the plant look like if it was in this type of condition? And then also being able to talk about beneficial organisms, so like what's going on in the space around them and what's actually helping those plants grow, and then they would write about that. So they actually had this journaling activity, so again, bringing English arts into the garden space. And also this particular program is very centered on youth decision making. So it's all about the youth having the opportunity to do things and make those final decisions themselves. And these youth were from like 4 to 6 years old. Junior Master Gardeners. This program is a little bit higher in terms of the level of information, but also the children that are involved, or really the youth, are from ages 13 to 16. So they're developing gardening skills as the Seed to Salad program does. They're also learning a lot more about plants, so not just that you put a seed in the ground and it grows, but like what goes into that, how does it actually develop, like what does it look like on a cellular level even. Understanding the basics of ecology, so understanding that plants just don't grow magically, but they are part of a larger system. And that they're contributing to this to other organisms that are growing there, like in the soil, whether they be animals, the birds, whatever. Also learning just the basics of designing a garden. So why do you plant anything where you plant it? You know, making sure the soil is correct, so doing site assessments, but also knowing that there's a lot of shade in this area, or how can we make it more beautiful? And up here is actually a picture of one of the bean plants that they were growing for one of the Junior Master Gardener classes. They also engage in a lot of hands-on activities. So you can actually see down here is not one of the Junior Master Gardener classes, but I like to pull a lot of those same kinds of learning objectives from the Junior Master's Gardener programs into my one-off lessons. But this was actually at a day camp that I visited with another Master Gardener volunteer. They were using a lot of those skills as well. So there are a lot of challenges in planning these programs, because a lot of them were made for entire academic years, and I had to fit them into a few weeks of the summer. So timing was a really big challenge within that. Also, another challenge with timing was being able to make sure that I could adapt a lot when people would show up later than expected, or they came earlier than expected, and really having to think on my toes in that sense. There are also diverse age regions. So actually in this particular group here that is pictured, there's people from like 12 to 17, and that's like a really large age group. And even though we broke it up between my supervisor and myself, it was still like a lot to handle. So actually trying to make things so that it felt like they were being challenged, the older individuals, but the younger people also didn't feel like they were being overexerted and that we were trying to teach them too much, and giving the older youth an opportunity to teach the younger youth. Also limited resources. We didn't always have all the things that we needed, like the seeds necessarily didn't come on time, or we didn't always have gloves, we didn't always have trowels, and also we didn't always have transportation to get people back and forth. So really having to be creative with that, but then also teaching the youth that, hey, even if you don't have all the things that you need, you can still grow things. So that was a learning experience for myself as well as the youth. Also language barriers, again with this group here, there was one student in particular who didn't know any English, so we actually had to work with her to figure out how to communicate different things to her, and had a student sometimes translate things to her, and it worked out fairly well. I think she seemed to be smiling a lot and I hope it's because she was learning. Okay, program outcomes. So throughout all of these programs, they were able to connect with the natural world, because so often we have these experiences where we feel as though we are one thing and nature is another. But these programs really allowed to bridge that gap. So down here, actually, in this picture below, there were a bunch of students there who didn't really want to touch soil, but then I was showing them like, oh no, the soil is great and there's nothing to be scared about, and there are worms, and what do you find in the soil, and really seeing them connect with something that was not human or not electronic. Learning how to plan, build, and maintain a garden space. If you see in this upper left photo right here, this is actually a group of students physically drawing out their gardens so that they know where things should go, and if anything didn't look right, they can just change it before actually putting something in the ground. So again, that idea of planning before you actually do something to not waste time or resources. Also bonding with their peers. So in this upper right corner, you see that there are two groups of sisters, and they ended up becoming friends throughout the course of the program. Also bridging the gap between people and food production, because right now so many people go to the grocery store or they get the groceries ordered to them because it's COVID. And the idea of being able to know where your food comes from and be able to grow it yourself is something that is very novel to a lot of people. And up here you actually see some of the girls that are eating these different things that grew in their own garden. And although you can see one of them is not particularly happy about all the things that we grew, they were able to eat a lot more vegetables than they normally would have because they grew them themselves. So there isn't this like inherent disgust for vegetables or anything, but really it's a matter of how connected are you to your food. So speaking of people, I would like to acknowledge those who were really important in my experiences during the summer. So I'm my supervisor, who's pictured on the left here, the farm stand manager, who I worked with every Friday and Saturday, the IT coordinator, who helped me a lot with advertising things, the Master Gardener volunteers, who taught me a lot about different gardening things that I myself didn't even know, the executive director, who was very instrumental in getting me back there every summer, the urban forestry educator, who taught me a lot about trees and just like woody plants generally, which was awesome, and the food systems nutrition team leader, because she was there to help me get to places physically, but also she had lessons that coincided with my garden-based learning programs. So thank you so much for your time.
Awesome.
Great job.
Good job, Martha.
Questions?
Lots of times for questions. We're over 9 minutes ahead.
Hi, if it's okay if I ask one, this is Chelsea.
Sure.
Chelsea.
Hi, Martha. Great job. That was a fantastic presentation.
Thank you.
And it's so great to see all the visuals that go along with all the work that you did over the summers. I was wondering, do you know how many other of these garden-based learning programs are going on linked throughout New York State with our cooperative extension?
I can't tell you an exact number, but I know that from my experience working with different people in different extensions, that they do their like own versions of garden-based learning programs. They also do them in schools, but I don't have like an exact number.
It seems like this would be a great program to sort of coordinate, because you have a lot of lessons learned that is with resilience and team-based learning and things that I could imagine expanding to a lot of communities and being part of that.
It's actually funny you say that, because right now I'm working with one of the people who is really, really important in my entire experience is Marcia Inshively, which Lily mentioned earlier, because I'm working with her to create a program called Seed to Supper, but with youth corps. So it's a program that already exists for adults, but this one is more focused on youth and also integrating concepts of social justice and voice and personal agency, as well as gardening and collaboration and leadership. So the idea of putting together all the things that I've learned and all the things that already exist in these awesome programs and just bringing them up to date and making sure that we're considering the intersectionality of these issues that so many different groups are facing within their own communities.
Wonderful. Well, these types of programs are going to be important now more than ever as communities are feeling fragmented. So thank you for all your work on this. This is great.
Thank you.
Yeah, just a quick question. Hi, Martha. It's Adriana. Hi. It's so great to see your work. Really, really proud of you. Great talk. I was wondering if you were explicitly, taking any measurable outcomes. I know it could be really hard across such a wide, age of children and teens you were working with, but were you kind of explicitly measuring any outcomes?
Yeah, so of the objectives that were present for each of those different programs, I wanted to do like different types of surveys and whatnot, and I did get a glimpse of that, and I had different students that would submit different comments that they had about the different programs that were happening, but I didn't have a lot of quantitative data. It was also really hard to follow up with all the groups because, again, like the chaos of the summer, working at a place that's really made for gardening, and so the best time to do gardening for that particular group, especially when people are actually available, is the summertime. So I had like so many different things going on that I didn't have like enough comprehensive data to put into a presentation. But it's definitely something that I definitely want to work on more when I continue this kind of work, especially with the work I'm doing now with the Seed to Supper group.
Cool, great, thanks.
Thank you.
Regina, your hand is raised. Go ahead. Unmute and go ahead.
Hello. Can you hear me?
Yeah, we can hear you.
Hi, sweetheart. I'm very, very proud of you for what you've done throughout the years, and I love you very much. I don't know too much about gardening, but I wanted to be online with this today for you, honey. Thank you.
Thank you so much, Godmother Regina.
Okay. Thanks a lot, Martha. Great job.
Thank you.
Our last talk today is from Yahi Zhang. Her concentration is in computational biology. After graduation from Cornell, she will attend Southwestern Medical Center for a PhD study. The talk today is entitled The CITF1 Mediated Regulatory Network of Copper Homeostasis in Arabidopsis thaliana.
All right, thank you, Mike, for the introduction. Hi, everyone. I'm Ihe. I'm very happy to share with you my honors thesis project in Dr. Olena Vatmanuk's lab, which is about the CITF1-mediated regulatory network of copper homeostasis in Arabidopsis thaliana. So copper is an important micronutrient which involves in many biological processes, including respiration, photosynthesis, and reproduction, so that it is essential for the growth and development of all organisms, including plants. Copper deficiency can reduce plant growth, fertility, and in acute cases will cause total crop failure. Meanwhile, excessive copper is also toxic. So the precise regulation of copper homeostasis is important. To identify novel transcription factors that regulate copper homeostasis, previous work at Dr. Vatmanuk's lab analyzed the responses of the flower transcriptome to copper deficiency by RNA-seq. And among the upregulated transcription factors, the expression of a previously uncharacterized member of BHLH family was upregulated to the highest level. So we characterize this transcription factor and name it CIF1. And here I'm gonna show some further investigation on how CIF1 works at protein level in addition to these transcriptional studies. Firstly, I tested whether CIF1 responses to copper availability at protein level. So I collected plant tissues from rosette leaves and roots under copper deficient versus copper sufficient conditions. And the total protein was separated into cytosolic fraction and the nuclear fraction to check its subcellular compartmentation. We can see the CIF1 protein is upregulated in cytosolic part under copper deficient conditions, which indicates CIF1 may have important function under copper deficiency. And if the copper is sufficient, the plant may want to remove CIF1 to shut down the copper deficiency response genes. So next, we wanted to check if CIF1 is under this kind of control at protein level when CIF1 is sufficient. And our hypothesis is CIF1 protein might be degraded under copper sufficient condition. So to further confirm if excessive copper can accelerate CIF1 turnover, CHX, which is a translation inhibitor, was used to block further protein production before copper treatment. So we can compare the degrading speed of the existing CIF1 protein. So based upon CHX treatment, the existing CIF1 was degraded much faster under excessive copper concentration compared to control condition, which does not have copper treatment. Therefore, we can confirm that the turnover rate of CIF1 could be accelerated under copper sufficient condition. Then we want to know what happened when copper is sufficient. It should be go through some degradation pathway. And one common degradation mechanism in cell was the proteasome-mediated pathway. So to test whether the proteasome degradation pathway was involved in CIF1 turnover, depending upon copper availability, MG132 was used. So I incubated C1 transgenic plants with MG132, which is a proteasome inhibitor. And the result shows C1 protein accumulated when treated with MG132. Or in other words, when the proteasome activity was inhibited, which means CIF1 was degraded by proteasome. So to summarize all these findings, CIF1 plays important roles when copper was deficient. And if the copper was sufficient, CIF1 will be degraded through the proteasome-mediated pathway. And here is a diagram of this working model. And I would like to express my gratitude to all my lab members and my mentor, Dr. Olena Vatmanuk, and my direct supervisor, Dr. Ruzhen Jia, and all the funding resources that supported my research. Thank you all for being here and listening, and I'm happy to take questions.
Rosemary has a question. No? Okay. Well, you showed up there. Any questions? Great job. Very good job. Boy.
Well done, everyone.
All right.
Well done.
All right.
Fantastic.
Thank you all.
Thanks, everyone. All of our students for doing a fantastic job under a very challenging situation. You did a fabulous, fabulous symposium.
Can you guys all put your video back on?
Yes, great to see you all. Okay. Any closing comments? Anyone?
I think we should thank Mike and Chris for allowing this to happen and advising everybody in their presentations and being great mentors, I think.
Indeed. Thank you both so much.
Thank you.
Thank you.
This is our favorite class to teach. It's great.
Indeed.
Yes.
And we hope to be in contact when the university decides what we're all going to do for graduation.
Right. I would note that at our high point, we had over 50 people zooming in to hear you all. So that was great. Thanks for everybody who attended, for all the great questions. Yeah, well done. Thanks.
Well done. Thanks, everyone. Cheers.
This has been a production of Cornell University, on the web at cornell.edu.